पंजा 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 ధినీం డోలాసురమర్దిని
ధనలక్ష్మి ధాన్యలక్ష్మి ధైర్యలక్ష్మి
ధినీం డోలాసురమర్దిని ధనలక్ష్మి ధాన్యలక్ష్మి ధైర్యలక్ష్మి విజయలక్ష్మి
ఆదిలక్ష్మి విద్యాలక్ష్మి గజలక్ష్మి సంతానలక్ష్మి అఖిలాండేశ్వరి చాముండేశ్వరి పాలయ మాం గౌ.
Now, I would like to call upon our visionary principal of VIPER, Dr. A. Ramesh, onto the stage to accompany our guest. Now, I would like to invite our guest of honor, principal of VIPER, Dr. A. Ramesh.
I request Varshini to present token of love and respect to Dr. A. Ramesh. I request Nandini to present token of love and respect to Dr. Y. Sridhar. I request Varshini to present token of love and respect to Dr. Ranga Namburi. I request Lolita to present token of love and respect to Dr. Krishna Devarakonda.
Light fades away darkness. It symbolizes knowledge and wisdom free will. To solemnize this special invitation, I would like to entreat our dignitaries on the dais to illuminate this lamp lighting session.
I request Mahati and Kavyasri to come onto the stage for prayer song.
महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि वसिष्ठवामदेवादिवन्दितमहागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि वसिष्ठवामदेवादिवन्दितमहागणपतिम् महादेवसुतम् महादेवसुतं गुरुगुहनुतं महादेवसुतं गुरुगुहनुतं मारकोटि प्रकाशं शान्तं महाकाव्यनाटकादिप्रियम् महाकाव्यनाटकादिप्रियं मूषिकवाहनमोदकाप्रियं महाकाव्यनाटकादिप्रियं मूषिकवाहनमोदकाप्रियं महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिम्
Thank you, Mahati and Kavya.
It's my honor to introduce an inspiring leader and a dedicated mentor. Our principal has fostered a culture center on students' learning and growth. With his vision, wisdom, and unwavering support, he continues to guide both students and staff towards constant improvement. Thank you, sir, for motivating us and shaping a community where success and learning thrive. Dr. Ramesh Alluri, a distinguished academician and a professor, principal of Vishnu Institute of Pharmaceutical Education and Research, distinguishedly known as VIPER, with an impressive career spanning nearly 27 years under the Sri Vishnu Education Society. He has been a pillar of excellence in pharmaceutical education. Dr. Ramesh completed his MPharm from Andhra University and earned his PhD from JNTU Hyderabad. Starting his journey as a lecturer, he steadily rose through the ranks to become a respected leader in academia. An active member of professional bodies like Association of Pharmaceutical Teachers of India and Indian Pharmaceutical Association. So I would like to welcome Sir to address today's gathering.
Distinguished guests on the dais, Dr. D.R. Krishna sir, Dr. Ranga Namburi sir, Dr. Sridhar, of the dais, Director and Principal of BVRIT, my professional colleagues, Sudhakaran, Aunab Srinivas, and all my staff members, Mujibuddin, and all the teachers of various institutions, and dear delegates, a warm welcome to all of you for this Aushadh 2K26. This is the 14th international conference, which is annually conducted, and we are blessed to have all the delegates from across the states and international participants involving in this conference. As you all know, Aushadh means health and medicine. So with all your sparkling minds, young minds here, with lots of academicians, and we have industrial specialists, healthcare sector involved in this conference, can definitely be the platform to enrich ourselves with the latest updates happening across the world. I'm very happy to share that VIPER has been an important institution in Telangana with the #1 private institution ranking of in NIRF and the only college with the drug testing administration approval with NAC A plus and NBA, etc. I'm happy to share that we have almost, to my knowledge, updated, we have around 829 delegates as of now, and still there are few people who are going to join us. So I once again thank all the principals of the Telangana for having faith on our institution and sending your delegates for enrichment. And I also wish to thank all the speakers here, the chief guests. We are very lucky and fortunate to have one of the distinguished personality in entire India who is known and even now in abroad, Dr. D.R.K. sir. Thank you so much for addressing our students now, and it means a lot to us, sir. And we also have a senior industrial person who came from US, and he is now going to deliver after the keynote address by Dr. D.R.K. sir. Thank you so much, sir. And I have my young friend who can rejuvenate all the young minds, Dr. Sridhar, who is with us. Thank you for sparing your valuable time despite your busy schedules. Thanks you all. And I would like to thank all my staff members, students, and most importantly, this talk would never end without the mention of all the delegates who came very far from the various city places and landed here. A warm welcome once again to all of you for this Aushadh 2K26. Thank you, one and all.
Thank you very much, sir. We are truly grateful for your guidance and support.
May I request Lolita to grace the moment? Words about distinguished guest.
Respected chief guest, esteemed faculty, and my dear friends. It's an honor to stand before you this morning. I'm Lolita from third year. It's a true privilege to introduce a leader who defines a modern pharmaceutical landscape, Dr. Y. Sridhar. Dr. Sridhar currently serves as a Vice President of Clinical Strategies at Aminal Pharmaceuticals. His career is a masterclass in combining deep scientific expertise with global leadership. His academic career is exceptional. After completing a B.Pharm and M.Pharm, he earned a PhD in clinical pharmacology and therapeutics from Nizam Institution of Medical Sciences. Always standing ahead of curve, he furthered his education with Executive MBI from IIM Calcutta and a specialization in AI machine learning from Boston University. With over 20 years of experience, Dr. Sridhar has a pivotal global roles at industry gains like Novartis, Hetero, and Workhard. From leading global clinical trials to heading International Pharmacovigilance Department, his work has been consistently centered in one core mission, which is patient safety. Please join me in welcoming leader Dr. Y. Sridhar to address the gathering. Thank you.
Good morning, everyone. Firstly, today I am unable to speak because my respected mentor, Dr. D.R. Krishna, is here. Because how many of you have thought of dreaming for an MBBS? Then Everyone. So I was also like that. One of the youngsters after my intermediate, I was only focusing for the MBBS. It was the person who guided me is Dr. D.R. Krishna to the right direction, and currently where I am because of him. Thank you very much, sir. On this occasion, I'll take this. So from that, B Pharmacy is a role model for us. It's not only for me, it's for various generations, which has been inspired because of his visionary leadership as academician, as well as his thought process in the drug development process. Hence, I am into the drug development process now. So saying this opportunity for expressing my gratitude to sir, and I express my sincere honor and privilege for the BVRIT team, their management, and Dr. Ramesh for uh, inviting me for this good college. And very few colleges in India, what I have seen is with this infrastructure and full-fledged staff and yeah, the overall educational professional curriculum which that brings up here. So saying this, and also I am thankful to the Dr. Anga sir because of with the seniors who on the formulation development, where the current digital era where we are transforming for the new drug development, the completely from the after biotechnology into now, I will be speaking more on the biochipron technology and AI driven tools in my keynote address, mainly on the other tools that we can use for the development of the new drugs, which are more promising for the next generation. So ultimately means a simple thing in this short message, which I want to give is how many want money in the life? Everyone is wants. But without education, money, is it possible? Right? Yes? Muji only will tell us. Right? So the main criteria here is the curriculum where we are in the academic institutes. That is more important to come into the industry. If we follow the curriculum syllabus that is framed by the PCA and everywhere, if we follow that very clearly, then we will have a good thought process into what sector because pharmaceutical sciences is a very vast area where one can select based on their ambition to one area. So if you see to means anything, the research or the innovation comes from academic institutes and research labs, whereas industry make it more commercialized manner of it. So you can understand that. So industry will not teach that time. We have to execute in the industry from what we learn from the academic institutes. Let's assume if we want to, because we are in the pharmacy, if we want to start a medical store, how many are ready to invest because we need money, right? How many you are ready for investing? We will go to another means 50 kilometers away from Narsapur to some other remote area. Do anybody invest with me? You want to invest in the center of the Narsapur or in the 50 kilometers away from the Narsapur? Center of the, why? ROI, return on investment, right? So we need to get good return of investments. If you are such thought process, do you think how many multinational pharmaceutical companies are there in India? Anyone can name it? Nobody knows. Novartis, J&J, GSK. So you name any top 20 international pharmaceutical companies, they are in India. Where they are located? Most of them in Hyderabad and Bangalore. And if you see top 10 or 20 IT companies, which are into life sciences and healthcare, can you name any IT companies? Want to reason from the students? We come to the academic institute so that we get that josh, motivation back to learning. There is no noise, right? So all top 20 pharmaceutical as well as IT companies, they are in India. Why they have chosen for India as a destination? or the global centers, GCC centers in India? Because of the cost? If it would have been because of the cost, it would have gone to China, Philippines, or many countries which are more economical than India. So when I remember when we were in Novartis has decided to come to Hyderabad because there were 42 different factors like climate, employee friendly, mainly the intellectuals, professionals, everything. So based on that, the Hyderabad as a destination, when in terms of climatic conditions also, means away from all the climatic conditions are non-disputed, employee friendly, happy to live place. So that way, Hyderabad is coming to the first place in the globally to the eastern side of the world after US. So hence these global centers of most of the companies are coming here and they are establishing. Who has to utilize those opportunities? Somebody from Japan, China will come here? No, right? So it is our advantage where we have to excel in our academic curriculum and also have more innovative thought process so that we can flourish, because pharmacy is a noble profession for the healthcare betterment of the healthcare for human lives. So if we consider that and do I was in Switzerland, Basel with Novartis. The rainwater, which is a river, and even the fountain water in the gardens is drinkable water, microbiologically tested. Is our own villages or our own towns, is the water or the cosmetics or the medicines that are being done, how safe they are we properly documenting those safety measures of ever? Because as per the statistics, the top 5 leading cause of death is because of the medicines, ADRs, because of the medicine. So hence there is a lot of big opportunity to invent new more promising molecules which will be more safer for a human race, and we will have better potent molecules in many unmet medical needs. So that future is you as a students, you have to transform that into a better life for all the human beings. Saying with this small message, thank you everyone once again. Thank you and management also for BVRT. Please focus on this. Thank you.
Thank you, sir. We are grateful for your inspiring words. May I request a huge round of applause?
Now, may I request T. Nandini to grace the moment with a few words about our distinguished guest.
Good morning to the dignitaries on the dais, off the dais, respected principal sir, faculty members, and all my dear friends. I am T. Nandini of second year B Pharmacy, here honorably introducing our guest, Dr. Ranganamburi Garu. Dr. Ranganamburi Garu is a seasoned pharmaceutical professional with over 35 years of experience in drug production, development, analytical methods, and regulatory compliances. He holds a PhD in pharmaceutical chemistry from Andhra University. Currently working as a pharmaceutical consultant, he specialized in validation and compliances, remediation projects. He has held senior leadership roles, including Director of Formulations Development at KVK Tech and Technical Officer at CIS Pharma. Dr. Ranganamburi Garu has extensive expertise in developing oral, injectables, nasal, and ophthalmic dosage forms, along with significant experience in ANDA, NDA fillings, and FDA regulatory process. He has contributed to multiple approved drug products, filed several patents, and continue to focus on innovative drugs delivery technologies. Now, I respectfully invite our guest, Dr. Ranganamburi Garu, on the podium to address the gathering.
Thank you.
Good morning to everyone, and especially let me say good morning to Dr. D. Ramakrishna. I'm so proud to tell the audience, the delegates, that he is my classmate, and he was academically very talented. He was a Shroff Medal medalist in our at that time in our B Form, and I'm so proud to be associating myself with him in this conference, Aushadh 2020, 2K26. Let me also thank Dr. Ramesh because Last time he gave me an opportunity, I think I spoke to a few of the students here, delegates here. Also, let me also make a mention of Dr. Sridhar. He is a very accomplished professional in the field of pharmacy. I just want to say a few words before, anyway, we are going to talk in our addresses, keynote addresses in the afternoon. Maybe Dr. DRK is going to give his chief guest lecture before that. I want to say a few things. Aushadh. What is Aushadh? I was just, briefly browsing, doing a lot of this after pharmaceutical studies, going into industry, initially to start with pharmaceutical analysis, for product development, filing ANDAs, filing NDAs in US, and of course I also used to work here in Hyderabad, and one factor that I just came to, it was very striking for me, was you are the talent. You are the talent, and that was the reason why Hyderabad was chosen by many pharmaceutical companies, including, he didn't mention that Amneal is also a multinational company because it has facilities all over the world. I just follow Amneal because of some other reasons. So here, you are the talent. And also this Aushadh, which is just browsing through, and what is the meaning of Aushadh? It's a medicine. Since when we know about Aushadh? So many, maybe thousands of years back also we know about Aushadh. But this Aushadh has transformed from Ayurvedic herbal medicines. I think I met one of your pharmacognosy, head of pharmacognosy here last time, study of crude drugs. So we used to depend mostly on crude drugs, herbal medicines, and even, I think even now also we depend on this Aushadh, this Chapa Mandu people call it as. That means it's working. Was it standardized? Was it not standardized? It's a different thing. But this Aushadh was working, is working. Now it is transforming to a level where all of us know that, with the AI, new medicines, new things. Sometimes it so happens that this, one thing that I just wanted to, I think this personalized medicines, I'm going to talk a little bit about that during our keynote address. So this personalized medicines was known for all of us in the beginning of maybe a few decades back. All of us know because they just prepare a decoction of a herb and give it to that individual. They don't make it that decoction for everybody. So Aushadh has taken such a tremendous transformation. Now this is a huge opportunity to the delegates here who have chosen the field of pharmacy, and you can choose a specific specialization as you move on, so that that gives you that happiness that you're part of a very novel profession of pharmacy. So let me also thank the management of Vishnu College of Pharmaceutical Sciences for giving me this opportunity, especially Dr. Ramesh. Thank him, and please, I have only one small request. Please pay attention to what is being talked, and then try to think yourself, because once you study in the classrooms, then what happens next? So you have to think to do what, how you can help the society in general, how you can transform yourself. And Dr. Sridhar was mentioning, are we here to make money? Yes, of course, I think you need some money, but at the same time, you're doing a very noble cause of helping people who need the help. With this, thank you so much.
Thank you, sir, for your valuable insights.
It is a great honor to welcome our distinguished chief guest, Professor Krishna R. Devarakonda. With over 3 decades in academia and 18 years of pharmaceutical industry, Professor Devarakonda is a titan in the field of clinical pharmacology. He currently serves as co-director at the University of Pacific and a founder of 6S Pharma Inc. His career is defined by leadership, having led roles as President, CEO, and Vice President of clinically developed of global recognized organizations. We are truly fortunate to have a leader who so perfectly blends innovation with service. Please join me in a huge round of applause, Professor
Krishna R. Devarakonda.
Good morning to you all. At the outset, let me thank Dr. Ramesh, principal of VIPER, for bringing me here. It's a great opportunity. And it's very nice to see, quite heartening to see many of my former students who were so young in those days, like Sunita and Bhikshapati and many others. I'm really happy that they still maintain their vigor and aptitude. And I'm also very happy to see one of my good friends and classmates, Dr. Rangaraju, who is sitting in the middle. And also happy to see some of those young friends who were very much associated with me back maybe 20 years ago or even longer, like Sridhar. It's quite heartening to hear to his words, kind words, that he still remembers a couple of times that I had the opportunity of interacting with him. During those times, he was slightly under depression because he missed his medical seat, because he was pretty competitive and very sharp and intelligent guy. And then it so happened, since I know his dad a little, and so I just interacted with him, and I didn't do much. I don't know what I did, but that's what he says. I did something. See, what really happens is any mentor or any teacher or any friend who is senior to you, if at all he is a well-wisher, he doesn't have to, or she doesn't have to give you so much. What all they have to do is just to make you realize the kind of potential you have inside you. So I spotted that potential in Sridhar, and today see what he is. He's a vice president of clinical development of one of multinational companies in the world, Amneal, and he has been doing extremely nice work and has been catching up with the most recent trends like AI, machine learning, etc. And he has been trying to integrate those with the clinical development. And off the record, we have been talking a little bit in the morning, and when he was telling the kind of work he's doing, I'm really thrilled. So when this can happen to a gentleman like Sridhar, why can't this happen to you? Any one of you, for that matter. And the second question I ask you is the same question that Sridhar asked. I will try to rephrase it. That is, how many of you want money? Now, that's the question he asked, right? So my question to you is, how many of you do not want money? How many of you do not want money? I think this is easier. Perhaps the number is not as big as those who really want to have money. But that is not what I want to say. If you want money and if you chase it, you want success, you chase it. You want power, you chase it. In all likelihood, you miss it. And when you miss it, the kind of disappointment, distress, is so much that it is unbearable, right? You want something badly and you don't get it. What happens? So for that, what I would suggest to you all is that, each one is cut for something. Each individual is unique. Just because somebody doesn't have good communication, abilities, don't discredit him or don't disregard him. Likewise, somebody is not really so great in academics, doesn't mean that he's not, fit for nothing. He's not. So everyone is having certain abilities or capabilities, and probably that individual doesn't know that he or she, what kind of properties or, talents or strengths they possess. And as a result, they are not able to, follow the path of success. So if you want to tread the path of success, what is extremely important is to be able to realize what your strengths are. At the same time, also, you should be able to realize what your weaknesses are. That is why, in most of the top level executive kind of interviews, what they ask you is, what are your strengths? What are your weaknesses? Now, if you keep on telling, I know pharmaceutical chemistry so well, I got first mark in pharmaceutical engineering. Now, that is not what strength is. Your strengths in terms of your personality, your weaknesses in terms of your personality, your behavioral skills, these are the ones. So once you have these, rest of the things are very easy. When I was doing my 10th grade, how much percentage I got in my 10th grade? I got 49 point something percent. I just missed my second class. So my dad thought, I'm fit for nothing. Okay, then coming to intermediate, I had the opportunity of meeting, like Sridhar said, touch with a couple of teachers. One of them, I did my intermediate with mathematics main, MPC in those days. And, I came in touch with a maths professor or teacher. And that gentleman, he spotted something in me. And until then, maths was a big scare to me. But then already first year intermediate was over and I was in second year and he was teaching us calculus. And he said, okay, this is the way you do math. And, first start doing, in a proper way, how maths has to be done. And try not to rote memorize things. Rote memorization means just by hearting things without understanding the concepts. So first start doing that. And then try to understand what are the basic concepts that are involved in deriving a particular equation or in solving a particular problem. And doing a math in the right way, how to put the margins, how to do the rough work and how to do the actual step in the math. So that has, changed a lot in me. And my chemistry teacher taught me how to read books. Until then, I was a Telugu medium guy and I was not able to utter in a single sentence without a mistake. But he encouraged me to read, you read all kinds of English books, English novels. So I started reading early Stanley Gardner's books, Perry Mason books, and then I started reading so many, any book that was coming onto me, thanks to him, he used to lend a lot of books. And that helped a lot. So don't think reading your pharmacy books or reading your class books alone is sufficient for you. That doesn't open your inside world. So in order for you to have that kind of aptitude, you should be able to have a lot of general reading skills. So please remember that. I don't say that it should be in English. Several great books are written in other languages as well. Several books subsequently I read are the translations of from Bengali, written by Rabindranath Tagore. These are great, treasures we have in this country. And, you keep reading general things. And fortunate you are, everything is just, at, a step away. Just sit in front of the computer and, just type in Gitanjali by Rabindranath Tagore. You will get English version, Bengali version, Telugu version, whatever version you want to, you want to have, you will get it. And that is certainly going to, broaden your horizon, and that will give you, how to take things in life. I was a teacher for, after completing my MPharm at Andhra University, way back in 1978, I did my BPharm in 76. 50 years we celebrated recently, completion of my undergrad, along with my friend Dr. Rangaraju. So we went back to our school, the Andhra University Pharmacy Department, and we had really great ball. We enjoyed quite thoroughly with our friends, particularly with the students, who are 50 years younger than us. So they were really astonished. Even, in fact, some of them were even jealous to see the kind of animosity and the jubilant, or vibrant mood we were all in. So, age has got nothing to do with. Of course, I'm not 65 plus. It doesn't mean that I'm, young inside me. So youth is just a state of mind. So remember that. Don't think that you are losing opportunities. Don't compare yourselves with somebody who is doing better, getting, 2 marks more than you and getting first rank. All these are just rubbish. So what exactly you are able to get, what you are able to understand, the concept, if you are able to understand, that is more than enough. See, after this, I'm going to give my keynote, at least that is the name given to it. Generally, I give a talk for about 1 hour, and Dr. Ramesh told me Sir, it is 20 minutes. I said, no, I can finish off in 20 minutes, even 10 minutes or 5 minutes, no problem. But, he is nice enough to give an extra 10 minutes, so 30 minutes. So the concept I have chosen today, it's going to be slightly more complex. And I also asked him what is the spectrum. The spectrum is first year B Pharms through M Pharms, and some of them are PhD, some of them are faculty with enormous amount of experience. And so it is quite possible. It is extremely difficult to satisfy, the entire range. It is something like, I hate sour taste. You go to a marriage party and, most of the dishes are made out of, tamarind. So I will have a lot of difficulty there. Even then, I try to take something and satisfy myself. So likewise, I strongly implore and I suggest to the undergrad students, particularly first, second, third year BPharm students, you don't have to understand everything that I'm going to talk today. Just to try to catch, what are those points which can be of some relevance to you in your future, that would be more than enough. So coming back to the first point, try not to chase anything in life. Don't go after money. Don't go after education. Don't go after wealth. Don't go after anything. They will automatically come to you. I never thought that I would step into industry corridor. And I was a teacher from the beginning. In 30 years, I taught in, Kakatiya University, 30 years. And I left Kakatiya University in 2007. And since 2007, it's more than 18 years, I have been working in the US and I was working in the industry. So what a paradigm shift it is, what a huge change it is. Not even in India, I moved to a different country. It's number one in pharma industry in the world, where I had to work and I did work and I didn't change any company. I worked with only one organization all through until I took voluntary retirement in 2023 and I started advising companies, startups and so on. And since, basically I'm a teacher, I always wanted to fall back into the teaching option. And I have been working as a director of one drug discovery development program in one of universities in California and likewise in Texas. And, we do advise industry, advise academic institutions. And my ultimate goal, where I am extremely comfortable, is in the presence of young minds, because this is where, I can say something which is like a sponge. You don't forget it. Your brains are like sponge. You are going to absorb whatever I say. And, you may not use it today. Probably 2 years down the lane or 5 years down the lane or 10 years down the lane. Okay, that day DRK came and, he said, don't chase anything. Anything in life, not necessarily in your academic career or professional career or any career or in your own life, just try to accept things, try to learn things with genuine curiosity, and that is certainly going to help. Since I'm going to talk a little more and my friends are also watch, so I would like to take it easy. I would like to continue my discussion with my keynote. Thank you so much for your patient listening. And once again, thank the management for giving me this gracious opportunity. Thank you.
It is our privilege to have you as our chief guest. Thank you, sir, for your inspiring words. May we have a grand round of applause.
Now, I request the respected dignitaries on the stage to release the version of abstract in CD format.
We would now like to felicitate our guest of honor, Dr. Y. Sridhar. I request the dignitaries on the stage to do the honors.
Now, we would like to felicitate our guest of honor, Dr. Ranga Namburi. I request the dignitaries on the dais to do the honors.
We would now like to felicitate our chief guest, Dr. Krishna Devarakonda. I request the dignitaries on the dais to do the honors.
Now, I request all the principals from the various colleges to collect the memento. Dr. M. Sunita Reddy, Principal of UCPS, JNTU Sultanpur. Onto the dais. Dr. D. Bikshapati, Principal and Director of TRR Group of Institution. Onto the dais. Dr. A. Srinivas Rao, Principal, Bhaskara Pharmacy of College. Onto the dais. Dr. N. Sivasubramanian, Principal, Gland Institute of Pharmaceutical Sciences. Onto the dais. Dr. M. Kiranmai, Principal and Professor, St. Paul's College of Pharmacy. Onto the dais. Muzibuddin, CEO, Clinisol. Onto the dais. I request Sudhakar sir to please come and collect the memento.
I would like to request our chief guest, Dr. Krishna Devarakonda, to present the keynote.
Do you have to check? You want to say something? Oh,
All of them again will be
sure,
leaving.
sure. No problem.
I request all the faculty members from various college and VIPER also to come onto the dais to join a photo.
సరే వాళ్ళందరూ పాపం కింద నిలబడమన్నారు కింద బాగానే వస్తుంది అలాగా బాగానే వస్తుంది ఇటు సైడ్ వచ్చింది లైవ్ వాడు లైవ్ జనాలు అందరూ చూస్తున్నారు
Thank you. No problem. All the best. Right. See you. ఉంటాను ఇక్కడ ఉంటాను హైదరాబాద్ అటు ఇటు వెళ్ళినా బట్ ఐ విల్ బి హియర్ ఆల్ ది బెస్ట్ can I have some water here by any chance? This one? Okay. Thank you. Pardon. Thank you.
I request everyone please be silent.
Okay. Are you able to hear me in the end? The back rows, are you able to hear? Okay, I would appreciate if you can take the seat instead of standing somewhere. And then your management has been kind enough to give us shawls, mementos, and also breakfast and coffee, probably even lunch. All those things do not matter to me. What matters to me is, please be silent and listen to whatever little that you can take from here. Okay? And keep your smartphones, if they are really smart, for some time to rest. And as I said in the beginning, whatever little that you can capture from this talk, you capture. And as I said, several of the things or concepts are probably a way above, and many of them are very, very, at par, and you can understand everything. Most of these things you might have learned in your, basic science programs in the past. And what I'm going to talk today, one of our guests today, Dr. Rangaraju, mentioned something about, personalized medicine or individualized medicine. Now it is called more broadly as precision medicine. Okay. So what lays foundation to this so-called precision medicine? And that is the concept today. You know very well that, we have been taking all kinds of treatment for various diseases or disorders from physicians. And all of us probably, are having the same kind of makeup, in the sense, you are two individuals from your class, say BPharm second year, you both look almost the same. You are coming probably from the same village, attending to the same college, eating the same kind of food. And just imagine that you have some problem, both of you are having the same problem, and you go to the doctor prescribes some medication, and the way you respond to the medication perhaps is not the same. It is also possible that one of you may develop serious adverse reactions, and one may have excellent relief, the other may not have any relief at all for the given dose. So what is happening here? Have you ever given thought to it? But moving forward, you are going to understand as you go deeper and deeper towards understanding of the knowledge of pharmacology, you are going to see, what is happening. So is it moving? It's not moving. Okay. Shall I say next slide? Maybe if that is easier. So this is a concept, and you are seeing in the title slide, which is a little hazy. You are going to understand that better. And this slide is even worse. Don't try to read it. The titles part of the titles have been encrypted. You cannot read them. But the story is very simple. This is a story. Pay your attention and understand what it is. This was published in 2006 in one of the most popular, highly advanced journals in medicine called Lancet. I think many of you probably are aware what Lancet is. Lancet is a very standard, high quality, high caliber journal, medical journal. It was published in that. This is about a story of a mom who gave birth to a baby, male baby, and the baby is quite healthy, normal delivery, PV. And then during the time of delivery, the mother had to receive some kind of very minor surgery. And the baby was born, and the mom was having pain, post-op pain. Would you mind closing it down? Closing the door, please. Yeah. So the mom was having a little bit of pain, and so the doctor prescribed a drug called codeine phosphate. So you know codeine is an alkaloid, which is an analog of morphine. And codeine itself is not an analgesic. Analgesic means painkiller. And so, but why the doctor prescribed codeine in certain parts of the world, like Western part of the world, it is very common to prescribe codeine for mild pain, because it's not the codeine that acts, but codeine gets converted inside the body into morphine, an important metabolite. And the process is called demethylation, and demethyl codeine is morphine. And this particular morphine causes relief from the pain. So the rate at which the metabolism or conversion of codeine into morphine takes place, is probably not the same in everyone. So what happened is the baby was born, and the baby is being fed by the mom right from the day of birth. And day 10, the baby developed, gray coloration. And day 11, it was serious. She had respiratory problem. And day 13, the baby died. So it's very abnormal. The baby was looking normal, was born without any problem. And after that, the mom was feeding him, because it's a male baby. And day 12, the baby died. And when they investigated what could have gone wrong, they tried to determine the levels of morphine in the mother's milk and also in the blood of the baby. And what they found was the morphine levels were at least 10 to 50 times greater in the milk. So it means that the mom has been converting codeine extremely rapidly, large quantities, and the levels of morphine are so high, and the baby got intoxicated and died. But the dose prescribed by the doctor was so normal, very, very normal dose. But at that dose itself, the amount of morphine produced was very high. So this is the beginning of the story. Can I have the next slide, please? And we will see what could have resulted in this kind of problem. So now go back into the history quickly. I think all of you know this guy, Gregor Mendel. Does it ring any bells, Gregor Mendel? Now, the man who came out with the laws of genetics. Okay, so you know that. And he was a Catholic priest, and subsequently he was given the name Father of Genetics. And he did most of his work during 1850 to 1865 or so. And he worked on almost 25,000 of plants. And he was trying to pollinate them and trying to identify the colors of the flowers. And based on that, he came out with the laws of genetics. And he didn't isolate any genes, and he didn't know what genes looked like. But based on the traits, based on the looks of the offsprings, he was able to come out with certain laws, and these are called as the laws of genetics. And eventually, he gave a very bold statement. And what he says is that my scientific studies have afforded me great gratification, and I'm convinced that it will not be long before the whole world acknowledges the results of my work. So this he said, almost in 1850s. And then can I have the next one? And what you look at here is, these are the laws. I think many of you must have read in your biology what happens when, pollinate, one particular flower with another flower. And you know about the genes, and you also know about the alleles. So every trait or every quality that you are receiving from your parents, it is obviously because of the genes, and each gene is comprising of is a pair of alleles, and one allele is coming from dad and another allele is coming from the mom. And based on the nature of domination, one of the genes is called as a dominant allele and the other one is called as a recessive allele. All of you know that? Yes or no? First year B forms, you know that. So why is it so important? You took so much trouble in bringing me here and I'm teaching you basics of genetics. So there is a reason for that. So if you look at the parental generation, where both the alleles are dominant in one plant and both are recessive in one plant, when you pollinate these two, the offspring, is going to be nothing but the dominant variety, that is, pink flowers. But the genes inside are different. One of them is dominant and the other one can be recessive. And when these are pollinated, you are going to have, these in different ratios. One dominant, two dominant in one particular flower, in one dominant and one recessive in another flower, one dominant and one recessive in another flower, and two recessive in the fourth one. So 1:2:1. This is pretty much like a very important observation. Same thing happens. Every quality that is heritable in us, if you look at your eyes, your mom has brown eyes and the dad has, say blue eyes, one of them is a dominant character. Your mom is tall and dad is short, the tallness is a dominant character and the shortness is a recessive character. Therefore, it is possible that you are going to be taller in the same ratio, 1:2:1 ratio. And the same analogy can be applied. Can I have the next one? Can be applied to other characters. One more. So can be applied to all kinds of traits. For example, some people suffer from high cholesterol, high LDL cholesterol, which can lead to heart-related problems, angina, and then eventually it can cause heart attacks. And the expression of LDL cholesterol is also one of the genetic traits or characters, and that is also expressed in the same way in the offspring. So every character that is possible or responsible for leading to a certain disease condition can be inherited or can be transmitted through generations. Can I have the next one? I'm trying to skip some of those slides which are not very relevant. Next one. So this is about the cell. All of you, no, go back once, please. Yeah. So this is the cell. Cell has got chromosomes in the nucleus. You all know that. How many pairs of nucleus that a human body has? Human cell has 23 pairs. And 22 are somatic chromosomes and one pair is called sex chromosomes. And sex chromosome, you have X and Y. And in case of females, you have got two X chromosomes. And in case of males, you have got one X and one Y. So why are we talking about chromosomes? Because chromosomes are the seats of the genes. They are nothing but genes, a DNA molecule and a certain portion of a DNA molecule, which has the ability of transcribing, the proteins. It can be called as a gene. And how many genes do we have? Human system has, thanks to the Human Genome Project, it was discovered that we have about 25,000, approximately 19 to 20,000 genes, and these genes are able to express proteins, and all our activities, they revolve around these proteins, which are expressed by the genes. Several of the diseases that we get are because of expression of the faulty genes, because there is some problem in the gene itself that will result in expression of a wrong protein. So, and this particular thread of the DNA, which is a double helical, all this, it is compressed together in the form of a spring called solenoid. And why am I telling all this? Go to the next slide, you will understand. Next one. You are going back. Forward, yeah, stop there. So these are the 23 pairs of chromosomes. And the last one in the blue box is X and Y, the sex chromosomes. All the rest are called autosomes, and these are responsible for somatic genes. Okay, and these genes, one of them, one of the chromosomes you are receiving from mom and the other one from dad, I mean the genes from mom and dad respectively, you can call them homozygous if both are the same kind of alleles, and if they are different, you call heterozygous. And again, if they are recessive, homozygous, these are different kinds of combinations we get from our parents. And these are going to give us, certain traits. Okay, how are they again relevant from the point of view of a disease? Now, if you look at one particular disease, many of you must have heard a disease called a respiratory disease called cystic fibrosis. Have you heard that name, cystic fibrosis? It is a disease which is seen as a lung-related disease where the baby dies very young, and this doesn't have a proper cure so far, and is a genetic disorder. And this particular disease happens because of a mutation in one of the genes located in chromosome 7. It is like, I live in Attapur and this is my, flat number, this is my street number. Likewise, every gene has an address, and the cystic fibrosis related gene is resident on chromosome 7. Likewise, you have sickle cell anemia, and it is there on chromosome 11, the gene corresponding to it. And in hemophilia, it is located in the X chromosome. That is why it is transmitted by women. So, it is coming from the X chromosome. Can I have the next one? Next slide, please. Further. Okay. So, in addition to this, so this is how the chromosomes are located, and that is the, the DNA thread, and the DNA is nicely wound. Okay, it is wrapped over a basic protein called histone. Histone are ball-like structures you are able to see. It's a protein. It is a strongly alkaline protein, and it keeps the DNA bound together. All this forms a compact mass. And it undergoes, the DNA molecule undergoes unwinding, and unwinding is required for the DNA to be effective or active. And interestingly, apart from the simple genetics, there is another context or concept called epigenetics. Have you heard the name epigenetics? Epigenetics means these are not transmitted. That means you're not going to see the genetic code changes. But what happens is, in the case of epigenetics, some of the external influences, we all think that if there is something wrong with my gene at the time of my birth, I'm going to get a certain disease. Not necessarily. Probably your genes are normal now, but because of your environmental effects, because of lack of proper diet and exposure to various chemicals in your lifetime, you can have changes happening inside your genome, and that could result in certain diseases. Okay, and that is called epigenetic pathway. And methylation is one, and some of the food materials that we consume, for example, broccoli, cabbage, cauliflower, some of these vegetables, they are very good source of methyl groups, and they are going to methylate your DNA. That in turn will alter the nature and function of the DNA. So to put it simply, your genes are responsible for certain disease conditions, and when they are mutated, and likewise, even if you don't have something wrong with your genes, it is possible that your environmental factors, various diets, chemicals, and so many other things, everything under the the sky can cause changes in your DNA. Next slide, please. So you could take a look at it. What can bring about this kind of epigenetic changes in your life? So if you are exposed to light, seasonal disease, exposed to COVID, it can cause certain genetic mutations. Likewise, any virus, smoking, drugs of abuse, like cocaine or marijuana and so on. Your financial status, why? Because the stress that you get. Likewise, exercise, your intestinal microbial flora, therapeutic drugs, the drugs that we are using for one disease can have an impact on your genes, can alter your genes, and your social interactions, psychological state, stress, the, what do you call that? The sandwiches that you eat consisting of, polycyclic hydrocarbons. Some of these can have a very serious impact chemically on your gene, and that in turn can result in diseases. And is that the same for everyone? Come back and remember the story I told. Second year B Farm, 2 girls or 2 boys coming from the same village, same height, everything same, eating same kind of food in the mess. But still, the way they respond to the treatment, the kind of diseases they get can be dependent on some of these epigenetic factors as well, in addition to the genetic factors. Can I have the next? So a concept called as pharmacogenetics. This was introduced by a person called Anno Gunther Motulsky. He is the one who coined this word. Any change in your gene can result in alterations in your in response of a drug. This is forward, this is backward. Okay, good, thank you. So if you look at this, certain specific genes, whenever they undergo change, this can result in alteration of a drug metabolism. So come back to the original story I told earlier, the codeine conversion into morphine. So that particular mother has some problem. And why that problem? She doesn't have the same kind of gene responsible for expression of a protein that is an enzyme which converts codeine into morphine. Therefore, her behavior is not the same. And so she is seen having that kind of poisoning. And the same concept, when you apply to the whole genome, we call it pharmacogenomics. So changes in your genome, multiple genes resulting in alteration of either drug metabolism or alterations in drug action. This particular concept in pharmacology is called as pharmacogenomics. So please don't forget, those of you who are studying pharmacology, pharmacogenomics is one of the most rapidly evolving fields of science, where you are going to study the influence of alterations in your genome. Genome is nothing but the entire bucket consisting of your genes is called genome. And any changes in one or two or three or multiple genes resulting in mutations which alter the protein expression, and this particular concept is called as pharmacogenomics. The same thing when you apply to the toxicology, it is called as toxicogenomics. Okay, I'm not going to go in depth into it. So why the genes will change? What kind of mutations that take place? What is a mutation? Any change in the gene. So why? What kind of change can you expect? Every gene is consisting of 4 nucleotide bases, right? ATGC, adenine, thymine, guanine, cytosine. So if only one particular nucleotide basis, so in place of thymine is removed, and in its place, there is appearance of cytosine, that's it. It is called a mutation. And how many mutations have taken place there? Only one mutation. Just thymine is replaced by cytosine. This is called a single point mutation. And likewise, it is possible that one or two bases can be removed. They simply disappear. They are being extracted out. And that also will result in change in the configuration of the gene. And alteration in the configuration of the gene results in alteration in its expression. That in turn will result in alteration in the functional activity of that particular protein. So I hope it is not highly complicated, very simple. Genes can undergo change only at one nucleotide base itself. And if it is more than 1%, then we call it as what we normally use the word polymorphism. And this particular polymorphism is the culprit why we are seeing so many people among us having different levels of metabolism of different drugs, the way we respond to the drugs. Okay, again, there is some problem. Okay. So how is this particular science important to us or useful to us? For predicting adverse drug reactions, for optimizing the drug dosage, and then improving the drug efficacy. So we're not going to use the same dose moving forward, probably 10 years down the lane. And we have already started. In fact, I had the opportunity of developing certain AI-based software wherein it is possible to make a prediction of what kind of change that you could expect and what kind of change that you can bring it in altering the dose based on the genotypic information. So this is an example. I'm not going to give a complicated picture since I understand that most of you would not be able to appreciate it. But if you look at this, what you are seeing is that every one of us is not the same. So some people metabolize a drug so much, the highest peak, and some people, they metabolize very little, and some people, they are metabolizing in between, and some people are not able to metabolize at all. These are called poor metabolizers. So just to take an example, one drug, and if we have got 300 people, we are giving the same dose to all the 300 people. Majority of us, 70% of us, are able to metabolize the drug normally. take the example of again codeine conversion into morphine. 70% of us are able to metabolize without problem. And then about 6 to 8% of us, they are poor metabolizers. In the sense, they cannot convert it into morphine. You give whatever dose of codeine you want, they are not able to convert it into morphine. And a very small percentage, 1 in 300, are able to metabolize quite excessively. So you just give a small dose of codeine and it is rapidly converted into almost morphine. So these are called rapid or ultra rapid metabolizers. So this is what we call as polymorphism. Among us, we have people. And what about different races? What about different ethnic groups? If you take these blood samples from American Caucasians, likewise, you take the blood from the African blacks, or you take from some other country like China or Taiwan, people having Mongoloid characteristics. Each one has different percentage of metabolism. And if you take India, it is the most complex situation. This is a very important point that all of you should remember. Since you are going to be involved in more and more drug discovery development programs in future, India is like a mixed fruit jam. Because some of us we have Dravidian origin, some we have got Aryan origin, some coming from East having the Mongoloid features. So we all got mixed up. And as a result, it is very difficult to say. In fact, starting in 1990, early 90s, when we started work in drug metabolism back in Warangal, we tried to characterize this. What is the extent of poor metabolism or extensive metabolism of various drugs? And we were getting very conflicting results depending on what kind of study samples you are taking. So these are some future challenges. So these are called as, based on that, if you have this information before, it is great, but otherwise not. Likewise, several other agents I'm not going to discuss in the paucity of time. But when I was doing my PhD way back in 1980 to 84, this is the time when I got did my PhD work. 84, I got my PhD. I was working in leprosy patients. I was working on drug-drug interactions. And the important drugs that were available in those days were dapsone, rifampicin, clofazimine, and other drugs. And the most commonly used drug was dapsone. And it was very interesting that one patient out of, I was going to the, almost various districts of Telangana and reaching out to the leprosy patients, collect blood samples and so on. So when we were doing these studies, what we noticed was 1 in 300 or maybe 1 in 100, 1% of the patient who is receiving normal dose of dapsone, after using it for about 10 days or so, the patient was coming back with severe exfoliative dermatitis. That means the entire skin is peeled off. And a very life-threatening situation. And sometimes, the patient was even dying. And, we were not knowing what the reason could be. And at that point of time, I'm talking about in the early 80s when I did my PhD, we didn't know the answers for that. But subsequently, as the science of pharmacogenomics has evolved, now we understand why a small percentage of people were extremely sensitive to the toxicity of dapsone. This is linked again to the dapsone metabolism. I'm not going to explain the entire slide, but simply it is like this. Dapsone, which is 4,4-diamino-diphenyl sulfone, it has 2 amine groups and it undergoes acetylation metabolism. And this particular drug, when you have poor acetylation status, it is converted into oxidative metabolite. And the amine group, when it is oxidized, you have chemists here. What happens? It is converted into nitroso compound. So these nitroso compounds are extremely reactive. They react with proteins and or peptides. And these complexes of protein, dapsone, peptides, or proteins, complexes, when they are handled by what we call HLA, and HLA is human leukocyte antigen that is there on every cell surface. When that comes in contact with the T lymphocytes, which are responsible for immunity, the T lymphocytes react overwhelmingly, producing a lot of cytokines. And these cytokines are extremely dangerous. They cause tissue damage, and they causes skin exfoliation. So only those patients who have got a specific type of human leukocyte antigen HLA-B*1301, only those. A small percentage, 1 to 2% of the patients carry this particular one. All the rest, 90% or 95%, they are safe. They don't have any problem. They are swallowing every day 100 milligrams of dapsone. But this 1%, they will have overwhelming toxicity to dapsone. So don't we care about that 1%? So 1 in 100 is 10 in 1,000 and 100 in 1 lakh. So we cannot ignore this kind of toxicity, not just for dapsone alone. You take any drug for that matter, has a similar kind of toxicity issues. Now coming back to this issue, we were talking about it, and pharmacogenomics, and then this is pharmacogenetics of morphine poisoning in breastfed neonate pudding, and this has brought about so much of commotion in the medical world. And since or just because that the mom was not genotyped, had they been able to genotype the mom, taking just a small blood smear and determine what her genotype is she very poor metabolizer or rapid metabolizer or ultra rapid metabolizer? The reason is this mom was carrying 2 alleles, and this is called CYP2D6, that is the name of the enzyme, and she carried 2 doses of the gene. There are 2 alleles, duplicate alleles, and they are ultra rapid. So she was metabolizing codeine into morphine very, very rapidly. And so whatever dose of codeine that was given to her, she was able to transform it into morphine, and that resulted in the death of the baby. So what is it you can do in that case? How do you know that whether a mom is having this problem or not? So that is where what we call the genotyping, like taking your blood sample, determining your cholesterol, will help the doctor what type of dose of atorvastatin to be given, 10 milligrams, 20 milligrams, or so on. Likewise, for every drug where it matters, not for all. For those drugs which have got high probability or high propensity of toxicity, you can certainly consider this. And as I said, she carried heterozygous CYP2D6 2A allele, 2 copies, and that resulted in the death. Okay, now I am going to skip many slides. The same concept is applicable for various drugs, and the sources, I will leave the slides here. Some of you, those who are interested, can read further or study further. And so this is one part. The adverse drug reactions are the toxicity. In 5 minutes, I will close. So in case of toxicity is not the same, not uniform across. And it is not only the dose, but also, the drug concentration versus the effect, the pharmacological effect. We take 500 milligrams of paracetamol or acetaminophen. All of us, after my lecture, have developed headache. Let us assume. All of you have developed headache after this lecture. What are you going to do? You are going to take 500 milligrams of paracetamol. Are you sure that all of you are going to get relief from the headache? No. Maybe your friends may say, you were seriously listening to DRK's talk. Okay, that could be one reason. But the second reason is probably the drug is not effective in you. Some of you are responding and some are not responding. This is the case with every drug that has been done clinical trials by my friend Dr. Sridhar, and they come out with what we call as the average doses. So the dose of paracetamol is 500 milligrams. Do we have a dose of paracetamol 400 milligrams in the market? No. We have 500 milligrams. Maybe now we have Dolo, 650 milligrams, and in some places 1000 milligrams, but not in between. And if you critically examine that, so some people respond even at low doses, and some people respond at normal doses. Some people may not respond even at higher doses. So some of you may not get relief from the headache after my talk, even with 2 tablets of paracetamol. Maybe 3 tablets can produce relief. So doesn't mean that you are something abnormal. You are normal, but the dose is not normal. And our clinical trials are so much skewed. We don't address this variability when we are developing drugs. And that is one of the areas where I was focusing in my research and in my advice to the pharmaceutical companies. How to conduct clinical trials more wisely taking all kinds of variability, particularly variability related to the metabolism or genetic gene-based metabolism. This is what I'm saying. 70% tolerate 500 milligrams, 20% experience toxicity, 10% may not respond at all. So in our general public, we have a scenario consisting of 4 types of people. Okay, 4 types of populations. So one population is this drug toxic but beneficial, drug toxic and but not beneficial, and drug not toxic, not beneficial, drug not toxic and beneficial. Which one do you want to be? Which basket do you want to be in? Only in one basket, that is drug not toxic and beneficial. Unfortunately, every drug when it is developed, we have all the 4 baskets. People with all kinds of conditions. So we don't want that. Toxic, not beneficial. Toxic but beneficial, no. And not toxic, not beneficial, no. But what we want is only toxic, not toxic and beneficial. And today the practice of medicine is like this. The population, different colors indicate different genetic statuses. They go to the doctor, and the doctor sees the patient as an average patient and then gives a standard treatment. And based on your fate or luck, either you may have improved outcome or no benefit or adverse effects dominating. And what is the practice tomorrow? I think Dr. Rangaraju may throw a little more light on that. And that is leading to the precision medicine. With 2 examples, I will close my talk. Listen. So you consider one example. This is an anti-asthma drug. Okay, it is not fish medicine. It is a drug that was developed. It is known as lebrikizumab. It is a biological drug, monoclonal antibody. And this particular drug, it acts only in cases where interleukin-13 is cytokine, which induces bronchial epithelial cells. That means it causes bronchospasm, respiratory spasm. And this particular compound, it is responsible for secretion of a particular protein called periostin. So I will simplify, don't get confused. When asthma patients come, 100 of them come, some of them are having this particular marker periostin, and some of them they don't. And when you give this particular drug, it is showing good response. But when you take those patients who are having periostin positive, the response is very high. So you are able to separate the response between those who don't have periostin and those who have periostin. So what does it mean? So those people who are able to secrete periostin, this drug is active, and those who don't, the drug is not active. So why do you unnecessarily waste this drug by giving it to people who don't express periostin? So this was very clearly shown here in the clinical trial that people who are positive for periostin, they respond better. And what is that you are doing here? You are using precisely a particular drug that acts only in a certain condition where it is positive. I will give even a better example. And with this, I will close my talk today. This is an academic story. Please remember that great research happens in academic institutions. And why can't it be in VIPER? Can you tell one of the most popular anti-cancer drug that was discovered in recent times for treating a blood cancer called CML, chronic myelogenous leukemia? It is a deadly disease. The average lifespan after diagnosing CML is about 2 years, 6 months to 2 to 3 years maximum. And people were not knowing the real reason. So this is the story of a PhD student or a student and a mentor. So this guy here, his name is Peter Nowell, and the other guy is David Hungerford. So these two, they were working in University Pennsylvania in Philadelphia. Hello, University of Pennsylvania in Philadelphia. This particular group, they were working on developing anti-cancer drugs. And this gentleman, the professor, did not have the skill of observing chromosomes under microscopes. So this was in the year of 1950s. So they didn't know what is causing CML. And at that point, he hired a PhD student, Hungerford. He is an expert in identifying chromosomes. And when he observed under microscope, all the patients suffering from CML, they were having a very short chromosome. So why this chromosome is seen in the blood picture of these patients is, chromosome 9 and chromosome 22, these are two normal chromosomes present in every individual. And in these patients, what is happening is these two chromosomes are cut. Nobody is cutting, they are cut. And then after getting cut, these two are coming, the two are getting fused. The small pieces of chromosomes, one coming from 9 and another coming from 22, these two are coming, they are fused and they are forming a new chromosome. So this is a new chromosome, short chromosome. And all the patients suffering from CML, chronic myelogenous leukemia, are showing this short chromosome. This is a great discovery. All the patients suffering from this, they were showing this in a short chromosome. And since it was discovered in the city of Philadelphia, it is called Philadelphia chromosome. Just Google it after going home. Philadelphia chromosome, and you will get the entire story and how it evolved. And then once this was identified, and this particular short chromosome is expressing an enzyme, tyrosine kinase, in large quantities, and that is causing cancer, CML. So now we know what is causing cancer. So the next story is the drug. And the guy who is sitting in the top, Dr. Drucker, and he took initiative in developing a new molecule, and it is called imatinib. So maybe, you can type in again imatinib or called Gleevec, and it is a super duper success. And the company made billions of dollars. That's a different issue. And it has revolutionized the treatment of CML. And it is effective only in those patients who are having this, Philadelphia chromosome. If you don't have Philadelphia chromosome, still having the CML, you will not respond to this drug. So that is the beauty of, the discovery. And then what happened was, this is just an example. This is an Indian guy living in the US. He was suffering from this condition, the CML. And, he started using, he is one of the participants in the clinical trial. And, he got relieved. He was initially given maximum 6 months to 3 years lifespan, and he is still alive. So 85 to 90% of the patients who are positive for Philadelphia chromosome, who received this treatment, they showed remarkable improvement in their life. And today, almost every cancer drug that is being discovered is based on, this kind of principle. Okay, another example, I'm not going to describe it due to lack of time. Breast cancer is one condition where, all breast cancers are not one and the same. There are breast cancers who, where the cells express a certain kind of protein, and we have a drug that can inhibit only that particular protein. Therefore, the woman suffering from breast cancer having that particular protein overexpressed because of the gene called HER2 gene, can be treated, and the drug's name is Herceptin 2. Now, if you ask anyone, every anti-cancer drug, more or less, is so specific, so personalized. My son is an oncologist. He is a pulmonary oncologist, lung cancer specialist. And if a patient comes, the first thing he does is, takes a blood sample, sends it to the genetic lab, and they will come out what kind of lung cancer it is, what kind of gene has been mutated. And then once he comes back and tells, this is a gene that is mutated, like KRAS or BRAF or whatever, or HER2, then immediately there is a drug already available. X and Y company has developed a drug that specifically inhibits this particular gene. So that is the beauty. And can't you call it precision medicine? So you are so precise. You can give it to only certain class of people or certain patients, and those patients get almost excellent remission within no time. So that is several examples. I'm not going to discuss all these. For example, this is for lung cancer. So many varieties of mutations have been identified, and how they were able to do it. So tomorrow, how the treatment is going to be like, you go to the doctor is going to take your genome analysis, environmental effects, and your geographic location, things like that. And thanks to the AI, you involve AI and identify which drug fits best to you. And that is why, or that is how you are going to personalize the treatment. And each one of them will receive one particular drug. And every one of them is going to have improvement as a result of this particular concept. So integration is the key word, buzzword in the future. You are going to get information from the medical imaging, genomic sequencing, which I just mentioned, health records, and then your wearable Fitbit or whatever you have, it gives a lot of information. This is connected to the mainframe, and all this is put together, and you make masala, and then based on that you are going to come out. AI is going to interface, and then you come out with an appropriate drug. And it is also possible that each one of us sometime in future are going to be given a barcode on our back. Take it easy. No. And once you go there, the doctor does the QR scanning, and he will tell, okay, you are having this and this mutation, therefore you should receive this and this drug. So don't be surprised if such a day comes. So thank you so much for your patient listening, and I hope. So since there is not much time between the session and lunch, I don't know whether there is any chance for questions. Okay, there is chance for 4 questions or 5 questions. Quick questions. Yeah.
My question is, it looks very clear, but how much is this being implemented in India and countries like where we are speaking about pricing, where we are speaking about healthcare is at the bottom. Whenever we have any conditions, we don't go and talk to doctors, but we still depend on some friends or something. But
Yeah.
the way you are looking at the medicine integration, AI, technology used by doctors, do you really think that doctors and pharmacists and patients are ready for this change in India? And what are the steps we need to think about it to take that active part?
It's an excellent question. In fact, if you ask me what is the present status, probably we are not there, but we are going to be there. What are you all doing? Why should I waste 1 hour of my time here talking, giving a lecture? Spending at least 5-6 hours of my day. If I'm not able to, kindle interest, stimulate interest at least in 10% of you, who is going to bring change in India? You think, somebody is going to, come in rockets and, bring changes in our minds? No, you will have to do it. So why, if you go to the US, where I have been working last 18 years in the industry, several of my colleagues in the company doing, extraordinary kind of work in oncology or in any kind of orphan diseases, rare diseases, etc., cardiovascular, and so on and so forth. Majority of them or many of them are from India. So where were they trained? Where are they coming from? So don't you work unless you are in the US? It's not necessary. So we are not inferior to anyone in the world. I think someone mentioned earlier, India has been picking up in various ways, and we have got extraordinary facilities. And it is very unfortunate if I say this at this point of time. Indian pharmaceutical company, I think many of you are aware of the statistics, or you can go back and take a look. World's 14%, India is located at the 14% or 14th place in terms of value of the pharma. Value-wise, India is at the 14, 1, 2, 3, 4. One is US. Like that, if you go down, we are at 14. Volume-wise, India is number 3. More than 60% of the world's vaccine production is happening in the country. And more than 50% of the generic drugs that we have, in the US, more than 50% are coming from the Indian industry. Your own Aurobindo, your own Dr. Reddy's, your own son, Lupin, so on and so forth. Every day I go to a doctor, anytime I go to doctor and take my prescription, I look at the label, I see Dr. Reddy's labs. I feel proud. Aurobindo or Lupin or some name. Almost 90% of the drugs that are being used, because generic drugs are cheaper there, branded drugs are very expensive, extremely expensive. And many of us are going to use only the generic drugs, and India is providing them. And many of us go another step ahead and say that India is the pharmacies of the world. enough. We had enough of that kind of distinction. We are supplying generic drugs to the entire world. But what is stopping you? What is coming in the way of developing new chemical entities? Don't we have good chemists here? Don't we have people who are capable of performing computer-aided drug designing? Can't we integrate AI in drug discovery? If you look at AI is now using tremendously huge amounts of drug discovery platforms. And then don't we have sufficient pharmacology knowledge? So what else is required for us to make a beginning? People say, we are poor, and I don't accept that. The other day, it seems, and I read somewhere in the newspaper, so 250 crores or something like that gold was bought in a single day. What is that festival called? Huh? Akshay Tidi. So people have tons and tons of money. We don't take it out. And if you take the history of companies like, I'm serious, you just go back and read, the history of Johnson Johnson. I went to the premise facility when I was in Europe for 3 years. I went to Belgium, a place called Antwerp, where Johnson Johnson company was established in Belgium. There, it is a single man's dream story. Same thing could have happened here too, like Dr. Reddy's and so on. People had vision. And the most important thing is, in those days, we didn't have enough, people with enough understanding and knowledge. But today we have. So I cannot accept that India cannot do it. Yeah, we have the potential, we have everything. And the only thing is, you should be prepared. And there are people like us, people like Dr. Rangaraj. Rangaraj, he's a very distinguished pharmaceutical manufacturer. Worked for nearly 30 years in the US, came back and is spending almost 50% of his time in a year. He doesn't have a lot of responsibilities. His daughters are married and all. They're happily settled. And he is just, trying to spend time to give what he knows to the students. What is required is just listen to them. Take just 1% of what they say. The same thing Sridhar. He's a senior guy. He doesn't have to come all the way from Ahmedabad just to give a talk. But try to catch, what exactly they want to say, and then try to take help from your faculty. They are also equally knowledgeable, and then keep moving forward. Now world is so closer. You are able to, watch K-pop or whatever, in no time. But why can't you do the same thing in case of your own field of science? Sorry. Yeah.
Yeah.
Sir, first of all, I'm super excited to meet you today because I know that DRK sir is having a lot of followers and fans, but I did not witness. So right in front of you, sir.
Oh, okay.
So I did not witness that, but today, because of Ramesh sir, I'm thankful to him to create such a wonderful platform to meet you personally
you.
and listen to you especially, because if teacher becomes a researcher, how beautifully one can express the concepts today we have witnessed, sir. Thank you so much for that. My question here is, so as you said about imatinib, I am excited too because we are working on it. And because though it has created the revolutionary move, now it is suffering from some ADRs. So to neutralize that ADRs, we are working to neutralize the ADR of imatinib with some phytomolecules like thymoquinone and also, sir, but some ambiguity is there with imatinib farm therapeutic action and ADR, sir, especially with respect to depression. In
Right.
some papers, they notified that it may cause depression on long-term use. In some papers, they notified in some countries they're using it for treatment of depression.
Okay.
So anything about this?
Yeah, in fact, that's really a very interesting point. And it's not just imatinib. Every molecule, it binds to certain receptors, right? If you look at the pharmacology, the receptor or enzyme or carrier transporters, it has a very unique way of interacting with the targets, we call target proteins, and produce the pharmacological effect. But we also have to remember that every drug, has got more than one kind of receptor or one kind of target. That is why in the development of a new molecule drug discovery program, we do on-target as well as off-target efforts. If you take antihistamine, for example, antihistamines, they reduce the histamine release and produce antihistaminic effect and, relieves from allergy. The same antihistamines, they have multiple other receptor activities. They can cause sedation. Some drugs produce good sedation, like Benadryl, or some of the older like chlorpheniramine, etc. They cause sedation as well. And subsequently, they try to reduce that part. So that particular activity has been reduced. Same with imatinib. It is possible that depending on how it is administered or what kind of receptors involved, there is a possibility that it can cause either antidepressant effect or, can cause, pro-depressant effects. I need to look more deeply into that. And that is the space where today we are focusing. I have been now advising a couple of startup companies. They are not working on new molecules because some of these are Indian companies and they don't have so much of money for developing new chemical entities. But what they are doing is they're using already available drug, like same imatinib, we are working on another aspect. And this particular, it is called repurposing or reprofiling. We are going to make use based on the route of administration or mechanism of action. You can modulate the activity and get a different action. Like aspirin, apart from being analgesic, anti-inflammatory, it is used as an antiplatelet drug. So same way here too. So I think we are good. Now it is time. I might have other speakers. I cannot take more time. Thank you very much once again.
Thank you, sir, for your valuable insights. It was really inspiring to all of us. Now, I request our principal, chief guest, and guest of honor to felicitate Dr. Dasari Varun, principal of Pullareddy Institute of Pharmacy.
Dr. Sudhakaran Raj, Principal, Vijay College of Pharmacy, onto the dais. Dr. Kantlam, Principal, Brilliant College of Pharmacy.
It's time for a quick refreshment. Please join us after 10 minutes. Make sure you assemble in the auditorium as soon as possible, within 5 minutes.
Sir,
I request everyone please assemble the order within 5 minutes after the tea break, okay? And also followed by the plenary sessions.
I request all the delegates to get seated in the audi. I request all the volunteers to bring down all the delegates into the audi. Dear volunteers, please become alert and bring all the delegates into the audi. Sir, ఇప్పుడే రావాలా Volunteers, please gather all the delegates into the audi. I request all the students to occupy the front seats first. I request all the delegates to occupy the front rows first. ఇవ్వలేదు తీసుకోవాలి పెద్ద ఇస్తుంటే
Everyone, please call your friends as soon as possible. We'll be starting our next session. I request all the students who are at the back, please occupy the front seats.
Plenary lecture by Dr. Ranganamburi.
ఇక్కడ పెడతావు లేకపోతే ఇక్కడ ఇక్కడ అక్కడ పడిపోతుంది అది ఇక్కడ పెట్టు రెండిట్లకి వెళ్ళిపోతుందా ఒకదానికి పెట్టి సరిపోతుంది కదా ఆన్ అండ్ ఆఫ్ ఉందా ఆన్ అండ్ ఆఫ్ అక్కడ తర్వాత అక్కడ ఆన్ చేస్తావు కదా అక్కడికి వెళ్ళిన తర్వాత 20 minutes, sir. నాకు 15 నిమిషాలు ఆగొట్టేస్తా 15 నిమిషాలు ఆగొట్టేస్తా ఎక్కడ ల్యాప్టాప్ కాదు నేను పడినా మా మీదేది నేను నా ల్యాప్టాప్ బ్యాగ్లో ఉండిపోయాను అదే అయితే అక్కడ నీకు ఇది స్లైడ్స్ నువ్వు మారుస్తావా పర్వాలేదులే స్లైడ్స్ నువ్వు చేస్తావా నేను చేయను
This is forward, sir. This is backward.
ఓకే
This is the pointer.
ఓకే థ్యాంక్స్ అమ్మా So I think let's make a beginning. I know that after the break we have few delegates here. That's good for me because I don't need to because very few people to assess me. So I don't have so many people delegates to assess me because that was good for me. Anyway, I think maybe we'll wait for a couple of minutes or start, sir. So your sir told me you can't take more than 15 minutes. Initially, he said you can take 25 minutes as per the program, but he reduced it further. So I just want to give some very few examples. Maybe lights are on. You want to put the lights off or? Yeah, it's fine, Andy. Yeah. Good afternoon, delegates. What I'll do is I'll just go through because I'm not from academic line, but mostly I have been spending my time in pharmaceutical industry, working in at different levels in pharma industry. I've been exposed to over-the-counter medicines. I've been exposed to generic prescription drug product development and filing NDAs. Filing NDAs basically 505(b)(2) applications. So those were the areas where I've been working in pharmaceutical industry. Now I'm an advisor, trying to help some local pharma companies and also maybe help Dr. Ramesh. If he calls me now and then, maybe I can come and maybe talk if the students like me. So here, I think Aushadh the 2K26, which is an international conference being organized, and it's really such a wonderful thing that the concept Aushadh 2K26. I don't know who coined that concept. It is a wonderful thing. It was very nicely, it's a modern thinking, and but at the same time associated with a known word, Aushadh. I think it's pharmaceutical sciences in the era of innovation, innovation through artificial intelligence, anyway, bridging research, industry and healthcare, and role of pharmacy students, because it's very important that what role pharmacy students can play and can be an excellent talent for pharmaceutical industries being located here and worldwide. Here, I was just going through, browsing through, I also use ChatGPT or maybe a AI-related tool is to understand. So I was trying to understand what was Aushadh. Definitely, I know what was Aushadh because people. I think the last maybe 3,000 years, people use the word Aushadh. I think it originated from India. So it's nothing but it is anything used to treat or cure illness, of course, in a different dosage form. And people, so widely used, Aushadh, our grandparents tell us, take the medicine. Maybe it's a more of a Hindi connotation, I'm not sure. See, I think I took some pictures how it got evolved, how this traditional medicine, Ayurvedic, homeopathic medicines got evolved into at the time Aushadh, and then the same Aushadh now with modern technology. And I think we know what is a modern puzzle, how people were making, take some herbs, crush them into. And then here, I just want to give you, to motivate you, I want to give you one small example, take about a minute or so. I used to work on, for my PhD program, I worked on Tifrosia purpurea, which is very wild, it was growing very wild. And we took the roots, and there was an indication that those roots, when crushed into powder, like a granular powder, put on a pond, and then sprinkle that powder, the fish used to come and bite, and then the fish used to get into a dazed condition to catch fish. That was one of the very old techniques. Now we have so many modern techniques people use to collect fish to feed us. So that was one of the very old technique. And then then for my PhD program, what was this ingredient responsible for making the fish into a dazed condition to collect easily? And that was a technique being used some maybe 40, 50, 100 years back. So we came out with, for my PhD program, it is so important for you here. Why I wanted to give you that example is it's so important for you to observe very carefully. So we separated into several fractions after extraction, and each fraction was being assessed for the kind of indication that was there to bring the fish into a dazed condition. So what was happening during the extraction process, we came to know there was an ingredient by name pongamol. Of course, we published those papers in international journals. That pongamol was so unstable during process of extraction, during chromatography, putting that into the column chromatography. I think all of us know, maybe in B Form, first year, second year, we get taught. So the compound was getting degraded, even in contact with silica gel, which we use as an adsorbent for TLC or for column chromatography. So the meaning is, in the process of extraction processes, we are losing the efficacy of the so-called effect that was known when it is in a crude form, but in a process of extraction. So this was happening, so which we were able to identify how to prevent that and how to isolate and how to purify and how to attribute that property to that particular ingredient. We were able to publish, isolated several compounds, identified. Even when the spectroscopy was not very well developed, we were able to do that. Of course, I was awarded my PhD program of the work that was done on Tifrosia purpurea, which is a herbal medicine. I think all of us know that at the time I was telling that when the Dr. DRK was talking about the precision medicine, that means the individual medicine. At the time, in the villages, when you have some problem, you go to the local homeopathy doctor or whomsoever it is. They make that and give it to, that means it was being given as an individual medicine in those years. I don't want, these are some of the herbs that people were using. So here the question is, say if we had to take one tablet 3 times a day or 4 times a day versus one tablet in a week versus one dose in 1 month. This is the kind of thinking, the newer talent, that's what we expect from you. This is how you have to think for you to develop and formulate the active pharmaceutical ingredients into a dosage form, whereby it becomes easy, convenient for the patient to take the dose instead of taking 4 times or every 6 hours. So if we can give once in a week, you're done with. Of course, targeting the receptors to give a better cure, especially for cancer drugs. Of course, this is a wonderful thing. Let me again thank the VIPER for giving this opportunity and for bringing the students together to make them understand what kind of things got evolved over a period of time and where we are now, where we were, and where we are now. So, see here, this conference, is very important for the delegates to understand what you carry, because after listening to Dr. DRK, I carried a lot of new things that even I was not familiar, because I was basically a formulator, and some of the things that it was very refreshing, some of the things that even I do not know. So listening to speaker and understanding and following it up, that's what we expect from the delegates. Is very important. That's what we expect. Of course, what is pharmacy? We all know. So what is formulation science? Is converting API to a dosage form, ensures stability, safety, and efficacy, because stability is very, very important, and also stability, how do you define stability of a molecule? Maybe about 10 years back, for blood pressure medication, what was happening is the formula was developed, everything was done, it was going into market, and some people were claiming, oh, we were not getting effect. What was happening was the active was undergoing decomposition in the formulation, and unfortunately, the analytical technique did not identify that the decomposition was occurring. So what has happened was, it was showing 100%, even if 75% of the drug was decomposed over a period of 3 months, 6 months. So please be aware that when we say it is stable, you need to apply the right stability indicating technique to ensure that the finished dosage form has the desired characteristics and assay. Here again, I want to bring another point, and I want to give some practical examples to make it faster. I think all of us know that ranitidine, Genentech, was the #1 prescribed product in the world for several decades. And all of a sudden, they came to know that there was an impurity by name nitrosamine impurities, which we were not knowing. And those impurities are highly carcinogenic in nature. And of course, it came out of FDA, and FDA put a lot of restrictions now, because even Hyderabad-based companies recalled their products because they were not meeting to those and nitrosamine impurities causing the side effects. Here, I just want to make some nanotechnology. Here, I am using the word nanotechnology because I want to focus here on that particular thing. VIPER has a nanotechnology center. Please, please, please use that center to do your own thinking. How do I enhance? How do I enhance the absorption? How do I make it more stable? How do I target my molecule at the desired receptor in order for it to be effective and at the same time little more safe as compared to giving a non-nanotechnology based formulation? I think we all know that there are tablets, capsules, syrups, inhalers, nose-based products, because a lot of people, to avoid the first pass metabolism, a lot of formulations are available in nose-based formulations. And there are so many products, including over-the-counter, including even the best medications that are available now in nose-based formulations. I think some of the nanotechnology, I just put they put the things here because nanotechnology is the technology center that's available where you can start taking some experimentations as part of your pharmacy curriculum to develop. I think all of us know anti-cancer compound, doxorubicin. When it is converted into a liposomal formulation, prior to that, it was having a lot of side effects, less availability at the receptor level. Now it was converted into a nanotechnology-based liposomal formulation, which has better effect, less side effects. So you have that facility, you have that center. Why don't you start thinking in terms of developing some of the active pharmaceutical ingredients whose safety is known, and then putting them into more safer, more stabler, reaching the receptor level in a better way. Here, I just want to, I gave this example, but again, I just want to. There was an ADHD drug, attention deficit hyperactivity drug, very widely used, mostly I think in US, to some extent in Europe. I'm not sure, I don't think we have that kind of issue here. I think they're not available also here, those dosage forms. So here, as a formulation scientist, as a formulator, as a pharmacy student, if you become a formulator, how to develop a dosage form which is patient friendly, having better efficacy, having better absorption, enhancing the absorption, putting it into the right form in order for it to be effective at the same time. Why I want to give this example was, see, a mom sends the children to the school. So when the student goes to the school, what happens? That boy or girl has this ADHD symptom. So the particular student has to take the medication. It's the effect is I think lasting about maybe 4 hours based on its elimination rate and absorption and those factors. So if it has only 4 hours of action, what will happen? The student went to the school, and then the mom has to again bring another dose, or the student has to take. So just imagine how the student attending class gets exposed, because all of us know that, because the society, this student is taking some drug, especially in a classroom environment. If somebody is taking some medication or medicine, what happens is they think that this guy is taking drugs and then attribute all other things. But whereas actually the student needs that medication in order for him to control himself or herself. So here, I just want to show you how, as a formulator, in fact, we filed an ANDA for this particular formulation, and then we went through, it's a gamut of studies. We do stability, we do first product development, we perform in vitro dissolutions. Then after stability, after in vitro dissolutions, then we give this dosage to the human volunteers, as I think maybe he knows about bioequivalence studies being conducted worldwide. So here, why I wanted to, as a formulator, what can I do to mitigate this society effect on the student or on the student mom, because the mom has to go? What could be done? Could you give this dose maybe after 4 hours? In the same dose, but what happens is the first drug release occurs immediately within 15-30 minutes. Then the action takes place. Then again, you have to give another dose. Instead of giving the second dose in another different a second tablet or second capsule, can you put into the same capsule or a tablet another dose that targets the delivery after 4 hours using appropriate technology, what we call it as a drug delivery technology? It's very important that please understand the drug delivery technology. Please try to understand the concept right upfront from BFARM 1, BFARM 2 level. So here there are two kinds of beads, pellets put into the same capsule. The student, when goes to the class, given, the action occurs for about 8-12 hours. Of course, subsequently, even 16-hour dosage form came. I just want to give you this example because this is how, as a pharmacy student, has to think, what can I do to improve the health of an individual patient, so that that person has the effect at the same time. All other considerations of society and those things do not play a role. I think they were put into a bead form, 2 different kinds or 3 different kinds into a capsule, and that capsule, when it is given, it gives the action either for 4 hour for some students, that means a single type of bead, 8 hours for some students, and maybe 16 hours for some population. So based on what is needed, you take that particular drug product. I use the word drug product like Aushadh, that is medicine, effective for 16 hours, effective for 8 hours, effective for 4 hours. I think this was already discussed. Okay, let me give you another example. Nanotechnology. Here, nanotechnology, all of us know there are different kinds of formulations that can be developed using nanotechnology. One simple technique is take the API, mill it into the desired particle size. So if the particle size is, say, 1 micron, it may have maybe 5% absorption. If you make that particle size to 400 micron, it may have 25% absorption. So what is needed is converting the API by milling process to convert that into a nanoparticle. And at the same time, one has to keep into consideration, when you convert the API, active pharmaceutical ingredient, you have to make sure you have to not only nano size, but ensure that the nanoparticles are stable, because if the nanoparticles again become agglomerates, the effect is lost. Absorption won't occur. So that means enhancing the absorption by milling technique. Of course, whatever medium, those details, if I go, it will take a lot of time. So I'm not going to get into, but at the same time, so enhancing the absorption by milling, which is nanotechnology, you have the center. So this is one technique. There are different techniques that are available with the nanotechnology. Okay, I want to give you another example. I think Dr. D.R. Krishna was also talking here. Oral peptide molecules. Oral peptide molecules like GLP-1 medicines. There are several GLP-1 medicines that are available in the market. Most of those medicines are available in an injectable form. As all of you know, injection means what happens if I have to take an injection and go to a doctor, oh, why did you come? What happens? When did you come? When we have to take that injection? Instead of all these things, if you can give a tablet dosage form, that's really good. So only semaglutide was the only molecule that was available in a tablet dosage form. Only one company developed semaglutide into a tablet dosage form. And unfortunately, the absorption is no more than 1%. No more than 1% of semaglutide, when it is taken orally, is available for it to have that weight loss / the anti-diabetic functions. So what can we do as a student? What can we do as a formulator? What can we do as a pharmacy student / delegate? How do we enhance that absorption from 1% to 5%, 10%? In fact, that was the most recent thinking that I was having. And then a week back, another company came out with, so this semaglutide was a very large peptide molecule. When it is taken orally, what happens? There are so many enzymes in the stomach, so many factors that prevent that absorption, first pass metabolism, that prevent that absorption and availability. No more than 1% was available. So I was thinking myself as a formulator, can we develop this, enhance this absorption beyond? I'll take another few more minutes, sir. So can we enhance this absorption, say 5%, instead of taking a 14 milligram large peptide molecule? The API itself is so expensive. Can I make that into a 1 milligram or 2 milligram tablet? That was the concept, that was the thinking. That's how we expect, as the new talent that is available here, that's how you have to start thinking to make it happen, that you are enhancing the absorption, whereby the cost of manufacturing of that pill comes down drastically, because these are very, very expensive. I don't know about the cost here, but they're prohibitively expensive in advanced countries. And what has happened was, I was thinking, and then this another company came, they came out with a small molecule. It's not a large molecule like, 37 amino acid molecule. It's a simple small molecule, and that for weight loss, and it's much more effective in a tablet dosage form, better absorption, no issues. So here, how the dynamics, not only you have to think what was already developed, what you could do, and if while doing so, if the newer versions have come, doing the work that was already performed, is it good? No. So always you have to upgrade yourself, understanding what is happening in the market, understanding what is happening in pharmaceutical field, understanding what you can do. You have to constantly think, otherwise what will happen is you end up with doing some work which has no commercial value. I think our friends Dr. D.R. Krishna and Dr. Sridhar, they were mentioning about money. Maybe I may have a slightly different opinion. I feel that that you have to professionally do well. Once you start doing professionally well, once you contribute positive things, absolutely sky is the limit. And another question was why India can do those things. As Dr. D.R.K. put it, absolutely we have the best talent. But did we come out with a new molecule entity or new molecule, like a new say individual molecule that is effective? But we are working. So it may take a little more time for us to come out with that kind of things. And in regard to personalized precision medicine, I think Dr. D.R.K. has put it rightly, that you need to have a better understanding now with artificial intelligence. Why should I take 650 milligram of acetaminophen when I can have the same effect with 400 milligram? Why should I take? Why should I have side effects of acetaminophen? Acetaminophen also has side effects, all of us know. Here, the another thing, I was mentioning that Aushadh prepared by ancient people in taking a mortar and pestle. Now there was some in between. There is a 3D printed tablet or 3D printed slug kind of dosage forms that people came out with, but of course mass production. If you have to produce 1 million, 10 million tablets using 3D technology, I don't think it is physically possible. But making a personalized medicine with the 3D printing, that is possible. I think the one medicine was already approved by FDA. I just don't want to get it. So the pharmacist, you have several roles. You can be a formulation scientist, you can be analytical chemist, you can be, our friend is the head of clinical division of a large pharma company. So you can be anything, provided you have the interest, provided you show that inclination right from B Pharm 1 level, so that I want to become this. I want to do something else. Even that API, when you start making this GLP, say 37 molecule peptide molecule compound, if you can make some slight improvement in process chemistry, that will have a tremendous improvement. Also, I was telling about, in the sense we are recently working on one this thing where we wanted to eliminate methylene dichloride. Normally in US we don't like, but still it is available in US, in Europe, in India, very widely used. Now we are working on, can I remove that MDC? That's my small topic that we are working currently. I just want to let you know, that's how we have to think, whether at the B Pharmacy level or maybe an experienced scientist or formulator like me, we have to think outside the thing, and then what improvement can we bring in order for us to do a better job, and in order for us to do a better, give a better health. I think AI driven, Dr. DRK has done a lot of things. I just put the, absolutely the AI. Let me give you a simple example. Unfortunately, that's happening, but at the same time, if we can upgrade our skills, it has nothing to do with the job loss. Let me tell you one simple example. Earlier, we were doing generic prescription drug products, means a lot of competition, especially from, in US. Some of the drugs you cannot really produce here, develop and market in US because there are a lot of restrictions. So we were doing that kind of medicines where developing the products, generic prescription drug products. And then in that process, I used to have about 20, 25 scientists reporting to me. So to prepare a scale-up protocol, that means we developed in a maybe a 5 liter this thing, or maybe 5 kilo batch size. So you cannot make it commercial at 5 liter or 5 kilo batch size. You have to produce maybe 1,000 kilo batch size. To write a scale-up protocol, we used to take two, three scientists and 1 or 2 regulatory, and a team of 4, 5 people take about a week, 10 days to write a protocol, scale-up protocol. But here with AI, absolutely, in 10, 15 minutes, it gives you the kind of draft protocol. Of course, you need to have knowledge whether the AI has given the right input or wrong input. But for that, you need to have the knowledge. That means you have to develop that knowledge in order for you to assess what the AI produced or given a protocol. In the sense, you need to understand, you need to have the knowledge. That is the knowledge that you have to acquire, so that AI will not come into your way to get a job. So this I already mentioned. See yeah. So I'll take 2 more minutes. Aushadh, medicine. It was there 3,000 years back. It was there about 40, 50 years back. It was there. It is there now. Absolutely. It is only the advancements in technology that is taking shape, and then understanding how the herbs were being treated. What do we need to do to make sure that the herbs have, especially all of you know that in US, the most recent, this thing is very widely used, is dietary supplement ingredients. How are they evolved? How are they developed? The dietary supplement ingredients, dietary supplement drug product, I don't use the word drug product, dietary supplement compounds are the dosage forms. So from where they came? They came from mostly from natural herbs and sometimes the isolated compounds. Say, for example, take curcuma. Curcuma is a pasupu. It has an active ingredient curcumin. When curcumin is isolated and given in a form, it has very minimal effect. Now, what do we need to do in order for us to get the desired effect? And at the same time, it is a known ingredient. It is a dietary supplement ingredient. There are no lot of regulatory quality standards and those things. Of course, now there are some standards. So not many regulatory requirements. Now, the sky is the limit to develop our natural herbs into dietary supplement products and put that product into market. And I think even in India is very widely used. So I just want to give that example. And now what was happening is this curcumin in nanotechnology, which is the center that you have, if one can start working on, that will help you how to put the curcumin with a better absorption when it is orally administered, because it should be available, it should get absorbed into the blood and then reach the receptor level in order for it to show the effect. So Aushadh, that is the thing that all of us know. So I just want to wrap up the things instead of elaborating a lot of things. So please, as student of pharmacy, you are the talent. You are the talent. That talent, if it can be if the talent is available, and that was the reason why world is looking at India as we have the best talent available as compared to China, as compared to Taiwan, as compared to several other countries. So these are the few practical examples I wanted to present to you so that you understand how formulation work is being performed worldwide. Thank you so much. Yeah. In the sense, I think because we have shortage of time, you can meet us. I'm here. You take my contact details. If you need some help, if you need some project-related formulation, project-related things, absolutely I have some knowledge because I've been working in pharmaceutical industries as a formulation scientist most of my time. Thank you so much.
Thank you, sir, for your valuable insights. Now we would like to felicitate our guest, Dr. Ramakrishna, Principal of Vageshwari College of Pharmacy. I request our Principal to do the honors.
He owns an industry. He's
Yeah, nice to meet you. And yeah. Yeah.
Thank you.
Let me know if you need some help. And
Definitely.
yeah, I have some for him.
Be taking the contact from him.
I like that.
Okay. The next session is followed by plenary lecture by Dr. Y. Sridhar.
Hello. Yeah.
Forward.
Backward. Mhm. Like, all right, no? Yeah, good afternoon. So again, I'm back on stage, but unfortunate for me is it is lunchtime. So, and okay, no? Good. If your interaction is over, then I'll quickly give the message what is game, because respected DRK sir and Ranga sir has already given a platform, because that is the opportunities that we are seeing for the future. With 50 years of experience, they have come here to give you the background very clearly structured way. I think at least if 30-40% of you will imbibe that, definitely your future is set. And my concept on the AI applications is secondary, but maybe you would have not born if you remember the issue of 2K when 1999, when the 2000 is coming 2K issues, where they said that all the systems will crash at once, so that everywhere. But that problem was also sorted out. So every problem will have a solution. Now coming up with the AI, I would not call it as artificial intelligence. I'll call it as assistant intelligence. It is helping in every field. So the last year when I went US, I have seen the Tesla and very thrilled to drive the Tesla. Very good futures. I was overwhelmed with the invention that has been done in this process. But after coming here in India, then my brother-in-law brought the Mahindra 9E, where when I compared with that with this has more features compared to the Tesla. So you can see the imagination. If one can put efforts even from India, it can be more better invention or innovation based on the current needs. So the automobile industry, which I was speaking, the AI giving auto braking systems and many other options. So similarly, in drug discovery and drug development process, because the entire pharmaceutical sciences is a big avenue where you have multiple options. From Bachelor of Science, you have many subjects, right? So every field is an opportunity, from pharmaceutical engineering, pharmaceutical yeah, chemistry, formulations, and also on the dispensing, many supply chain, drugstore, so many aspects of that. Everywhere it can be implemented. So if we think ourselves, where are the unmet medical needs? Or every product is safe in pregnant women? No, right? I can more means habituated Indians, we nod our heads this way, or this way, right? So please interact with us so that it will be, we can conclude it very fast. So in that context, if we need more safer products, because if we go anywhere in India, you can be seen many contaminations, or even the cosmetics left and right, everyone are using multiple cosmetics. Are they safe? The leads or the micro elements that are there, are they safe for humans? Even the material vigilance, whether the devices that we are using in the medical devices, are they safe? So everywhere, from the morning we were discussing, the water or the air or the surroundings, are they safe for human? Because we are here for the improvement of the healthcare as a profession. So saying this, I'll directly go into the, and all this whatever I am presenting is a public domain information, nothing related to my present company or the past company. It's my personal views and what I have worked in the academic institutes. So the basics of artificial or the assistant intelligence, there are multiple tools, like if somebody is aware of that, there are multiple softwares like brain inspired AI projects, chatbots, NLP, which is maximum being used. NLP is natural language process. And for more predictive analysis, even sir was telling, many on the genomic point also, many predictive analysis, data mining, or the neural network, LLMs and other things, and the other platforms. So with this, what can be developed and where the usage is there? Sorry. So if I go into the market, again, coming to the, it is not about money, because if you have Saraswati, Lakshmi will follow, the old statement. So the money is a matter where comes to for the every living aspect. But the knowledge is one which makes us to earn, or which makes us to get respect in the profession that way. So why I am coming with this is, in the drug development process, if you see the market analysis, from 87 USD billions, it is growing the exponentially high because of the introduction of this technology driven drug development process, where if you see the growth attributes for the other cyber security concepts or the data security, or from the attacks of the viruses, and also from the point of supporting the other businesses on the interactive games, real time. Similarly, in the pharmaceutical market, where from the drug discovery and the development process, the overall expectation is to reach more than, means 5,000 US million dollars. So the overall CAGR coming to the 32%, which is very high compared to the other businesses, petroleum industry or anything. That is the CAGR, compound annual growth rate, based on that. Hence, the major pharmaceutical or the IT companies are entering into the life sciences or the pharmaceutical business. It's not only in the product development, but also on the pharmaceutical services. So we have the opportunities there. So this is one of the market survey report where drug safety aspects, because even in precision medicine, we have the toxic effects of every product. So those concepts also, if we see, and emergent of regulatory guidelines for mandatory drug safety association reportings, because unlike a decade back, the reporting from the adverse events of this drug elicited effects is not there. However, now we have mandatory regulatory authorities from FDA, EMA, and as well as Indian regulations. So we have to report, and the information cascading from across the regulatory authority, that is one other reason, because we have now softwares available. On the same day, whatever we received for one product will be also reported to the FDA within 24 hours. So that is the timelines that they have given. So if you remember thalidomide issue, where more than 10,000 offsprings have been affected, and which was a big disaster which led to the ethics and the safety concept. Similarly, recently the nitrosamine concept in the various angiotensin converter or proton pump inhibitors, a excipient in the active formulation was causing the carcinoma. So the reporting pattern, when it has cascaded, the information that we receive in India is cascading to the other regions. This is called cross reporting. to the health authorities. And the same way, any other marketing region, if it is been reported any adverse event, that is been also received here. So the overall vigilance or the Vigiflow, which is developing in a database of the adverse events from the last decade. So we have an information on the product development and also on the adverse event for every class of drug. You imagine for the 3 to 4 decades. So this information will be helpful in the predictive analysis concepts and also the market growth for the so likewise, what AI can be leveraging in the opportunities that we have is on the first one is precision health that we the initial presentation sir was discussing, and the digital clinical trials which we are working, where from the randomization concepts, from also on the inclusion of the patient and also evaluation of the reports. So for to have an unbiased evaluation of the reports, it is not only means in one clinical site, it will not be evaluated. It is independent from any other country and a cardiologist for an ECG evaluation or any other radiologist for the other. So this is independent evaluation is giving more precision or unbiased evaluation. So those type, and you can see the digital diaries concept where we can have complete hospital related information or the what doctor has suggest you or a physician will be there in your palm. So that those app system based or pandemics. So there are many other concepts. So all these are the with the data like omics. You remember what is omics? Omics. Till now sir was telling na genomics, proteomics, metabolomics. So all those are the current concepts of omics. So you have to connect with that. And also many on the microbes or the electronic hospital records or the wearable devices where from this we can get this data. So the overall healthcare will be depending on the data that is reported. If we remember in COVID time, the remdesivir, which was used from all the healthcare professionals for the cure of mild to severe COVID cases. But WHO at the end, it has disqualified the product because it has do not have ample data. What is happened here is, it is not that underreporting, because of the critical situation, many physicians have not reported the adverse events or actual this thing. So that's the reason the data in these databases is insufficient to make informed decision. So what as a professional our role is to have this information documented to the appropriate health authorities or to the proper industry or every company has their own reporting system. Please report that, ensure that, okay, because we are Indians, we are used to adjust for whatever adverse event that developed by the product. That is not the concept. Or sometimes physicians are also afraid because some litigations may happen if we report the adverse event. That is not the case. So all the events, let's assume for one product, when we receive the list of adverse events, it will be evaluated. It may be evaluated as the listed or unlisted, or it is related or unrelated, or it is serious or non-serious. So based on that, health authority has given definitive structure. If it is already in the label or during development process, it is a listed event. And if it is a serious event, let's assume we have 10 serious events. So that doesn't mean that the what do you mean by serious, you know, right? Serious adverse events. Death is an untoward or extended hospitalization. There are 5 categories. So I will not go into the subject now at this moment. Serious is one event. When it occurs, it doesn't mean that the 10 event ensure that the product should be withdrawn from the market. It is that overall, let's assume if I have 2 lakhs of the product is being used, and out of this the statistics of 10 adverse event, how it has to be correlated. So overall the correlation will give a signal which is completely in a, whether it is really a signal which is hazardous for humans, or whether it is in some changes, whether in the prescribing information, warnings and precautions or contraindication section, it has not to be used in particular group of patients, those type of things. So don't go with a false assumption that every adverse event will lead to a product deformation or withdrawal of the product. So those type of misconceptions should be removed, but this reporting concept need to be Understood. So these are the various in healthcare concepts from the problem identification till the model and restriction. And conventional drug discovery pipeline, what we are seeing in the textbook, which is the basic foundation, where you go with serendipity or high throughput screening. That is a major concept. The new concept which has arisen, like sir has mentioned, very good, that is proteomics and genomics, from which the lead identification, optimization, again the complete process will be the same as the conventional process, but the technique of developing will be different. Similarly, if in sir has mentioned computer-aided drug discovery, when I was in UG, so that was the first concept where I was understanding from sir, the computer-aided drug discovery, where every moiety will have a pharmacophoric group and toxico, because of which the adverse event occurs. So it is to the tailor-made product which may give the superior outcome or therapeutic effects from the existing product. So those type of technology driven or the any more emerged technologies are there, those we have to be focused on for every one therapeutic area. So similarly, we have many other, as per regulations, there are many other process like after in vitro studies, you have to prove the CMC of the data, and preclinical studies. Again, the definitive schedule Y or clinical trial guidelines are there. Those we have to follow because that is mandatory for every industry. And also the safety pharmacological studies. Only unfortunate thing is when we were studying the PG in pharmacology and toxicology, it should be. Gradually, we ourselves has detected the toxicology, but there is a great demand for the toxicologist in the industry because all these are the battery of safety pharmacology studies that we are doing in the industry. Mandatory a toxicologist should sign because we have from the pharmacy profession, we have ignoring that, we are focusing only on the efficacy point of view. MVSC, veterinary sciences people are coming up with the toxicologist roles. So you can understand that is also equally important. We have to prove for every product this acute, chronic, and all this safety toxicology in reproductive toxicology studies, other toxicological studies. So those is also one of the important factor. And again comes to the human clinical trials. As you know, there are various phase 1 to phase 4 development studies and also phase 0 for the onco and the other the therapeutic areas. So this is the conventional model where the amount of timelines that used to take for complete drug development of one product. But there are other, like sir has mentioned, Ghazal and DRK sir, there are 505(b)(2) pathways and other incentive programs from the health authorities where a fast track approach in this drug development process can be possible. Hence, you have to focus on whether the safety is already established, then you have to go with the designing the concept whether it will fit into 505(b)(2) or the conventional process based on the moiety and the data that is available. So this is the conventional process as already you know. Yeah, the various battery of toxicity studies which I was mentioning, which is mandatory from the health authorities, including the complete ADMA studies. Sorry. So this funnel diagram, it is already you are aware of it. Yeah, only the critical thing which I am saying, the products which are developed and which are successful in the early phase of the development are going a failure rate at the end of phase 3. What is the primary factors? Because when many industry pharmaceutical companies were, so when we attribute like the factors which we were discussing DSK's presentation, that is one, and also one of the other important factor is the reasons attributed pharmacokinetics. The ADMA, the biopharmaceutics that we study in the B Pharm is more critical element in deciding and what product to be successful or not. So those ADMA factors from the is the 40% of which is the failure rate is because of that. So hence the initial studies, most of the gradually emergent phase 1 studies, we also combined with the pharmacokinetics so that it elicits the proper pharmacokinetics, then it is expected to provide the desired therapeutic effect. And the similar way, there are some animal toxicities, additional animal toxicity studies have been recommended by the health authorities. and also on the lack of efficacy, which is LOE is not at all acceptable, because we are developing this product for the efficacy of that desired therapeutic area or the problem. So if that is not there, around only 30% of which, and other reasons on the adverse events and the commercial reasons are very negligible. So this is the clinical trial complex, where to the challenges that we have from the paper based, when we emerged into the system based systems. If you see everything, the clinical systems that we use, there are multiple softwares. Even that is one of the career. It's like SAS Fidels. SAS is one. Oracle Argus, or the Medidata Rave. So there are multiple things. If you are somewhat tech oriented with professional knowledge, definitely those are the areas where the development of these tools for the clinical trials or the drug safety concepts, those you can have there. Also on the clinical supplies, it is not that routine shipments, it's not like, it is IATA approved product, means the specified temperature controlled shipment need to be, and those also need to be done by the professionals. So that is also, supply chain is also one of the big area. Central lab process, we have to have these NABL accredited labs to have this uniformity of the data. It's not. So all multi-centric or multi-country studies will be evaluated from the central lab, so that the reports are uniform, or the means the outcome will be more desirable as per the inclusion exclusion criteria. So each one area we is showing one expertise in where one can get their career opportunities, include the data management or the CRO concepts complete, or the project management if one is interested, or the business development concepts. So this is how we have evolved from the paper based to what is the CTMS, clinical trial management system, and multiple CROs. Now it is in a Power Blade cloud systems, AWS clouds and other Oracle cloud, where the data is stored for a larger. So the big data concepts have come, because we need large amount of data in order to evaluate desired decision making on the therapeutic effects. So saying this, we now have better understanding on the clinical trials, regulatory complexities, and also multiple protocol designs. If you are good at in writing, regulatory writing, or the clinical writing concepts, then we can even establish our career into the writing concept, because the understanding the regulations from EMA and FDA, certain products will be first line therapy in US and the second line therapy in Europe. Because of that challenges, where you have to incorporate into one product, because it should be a global study. So those type, either you do sub study or you combined it based on the problem, you have to come to a informed decision on that. So those type of understanding on the protocol designing or the essential documents for those trials, that is and how do we calculate, how do you collect the data, the sources of data collection, or the supply of the IP investigation products to the patients. These are also one of the concept, because in US, the CVS Pharma and other, where you can also get tie up and we can dispense the product. But similarly here also, whether the patient is receiving because the compliance of the product intake is also matters with the outcome of the study. So those type of close monitoring concepts also can be developed to see. So the overall collaboration of multiple area in terms of selection and completely without strategic planning of a long term, including the regulatory approvals or from the investigator sites and from the patients, everything should be collaborated, then only we will have a successful clinical trial outcome. So these are the various factors as we discussed, from the patient recruitment concepts and the regulatory requirements and regular reporting to the ethics committee and the regulator. So I have given a brief outer line, but there are multiple things in each phase, phase 1, phase 2, phase 3. Those maybe already it is included in JNTU syllabus, as we have included that. So you can cover there, but this is the highest means high level information where one can focus themselves based on their means genetic structure understanding on those concepts. Because I may be good at clinical subject, but I may not be good in business development concepts or any other concept. So you understand your competencies and deliver that to the maximum extent, like you chase that, you focus on that, definitely it will be a successful career. Saying this, I'll move into the some of the AI what in the public domain the major MNCs are working, mainly with the Pfizer is already working with the in vitro collaboration for the this machine learning and drug discovery process for some of the lateral ALS and the NASH diseases. And Novartis from my own company where we were working on multiple sclerosis. Fortunately, I was associated with this during that development initial stages, and it has collaborated with Microsoft and in silico medicine. These are the platforms for which various Alzheimer's and other multiple we are developing those concepts. And Sanofi is also working with on the Exentia and Oquin, and even the Roche is also working on this Oquin, and GSK is also working with the in sitro and extensions. These are the various platforms, and the most my favorite, the J&J is working with benevolent AI, which is giving more better understanding. Even the AstraZeneca is also working on that to give the more development discovery molecules. Saying this, in the AI structure, where how the target identification the candidate, how it is as service engine, the various options. Here also in the development process, there are multiple ways of the identifying the promising drug moiety, and also which elicits the more better efficacy and safer, like we need means zero toxicity, and predicting those efficacy based on the class of the drugs. If we have the one class of the drugs, we already understood that this is the what it elicits. So exclusively what are the adverse events or what are the untowards effects that is not required in this moiety should be focused and develop those for the additional better potent molecule. So likewise, in the same way, these are the various languages, like you may be knowing C language or SQL, PSQL. Similarly, in AI, there are multiple ways of developing, including some other Python or any other thing. So those concepts, basics of machine learning is one concept where you can use large data, complex data sets to identify the pattern, or the deep learning is where you can also identify the predict the 3D structure of the proteins with the deep learning concepts, or identify the novel drug delivery targets. And the NLP is more commonly used to extract the unstructured data into the structure, whereas also on the identify the potential these drug targets with their toxicity reports, and also computer vision to analyze the image to an. So now you are using the Gemini and ChatGPT, how the images are working out. So similarly, we can work with more but it is not that easy like a ChatGPT, because if you see the BT biotechnology, the amount of challenges that we have faced in developing those products in last 10 years. So similarly in AI also there are multiple challenges, though it is seems to be easy to develop, but there are multiple challenges that we are facing in order to make it more regulatory accepted to the human health. And one more concept is agentic AI, which is more better compared to the all these languages, which is giving more easy solutions, because it's like acts as an agent where whatever the intermediate between the two concepts, so that it will develop them the desired objective that we have selected. So these are the common tools and techniques that keep on using. So this is just I'm not going into the depth, because you can work on these concepts like in silico medicine or benevolent AI, and much more better concepts already started, but they are still at infant stage, because most of this because of the lack of data. Until unless we have more projects or more data into these tools, then these tools can develop into a more mature concepts which will elicit the desired. Even if you are entering into the ChatGPT, most of the times it will give false results, right? Because that was not used to it. But when you try to correct it, it will again correct. Similarly, these tools has to develop, get matured based on the data, actual data that is being provided. Hence, whatever the adverse events or the efficacy reports, it has to be published or it has to be reported to the health authority so that it can be data based. Because even the literature also, we will go into that, literature data also we will be documenting in the databases. So this is the concepts in depth what we were discussing on various atom wise how they are working. Here it comes, as we were discussing, the uh, death because of the disease is acceptable, but the serious events like death, unintended death because of the medicine with the treatment is not acceptable, because when we are in the pharmacy profession, we have to give the more safer products to the healthcare. So where there comes with this, the top 5 leading cause of death is adverse events globally. So this is the FDA FRS reports the gradually the increase in the serious and direct effects, which is untoward because of this medication. So there is a much more opportunity for every profession to have a more safer products into the healthcare. So that is more important. So for that, you can utilize this type of tools in from the AI or from the conventional drug development process, or sir was mentioning about drug repurposing. You can use for the same, or indigenous already available where the data is not established, but you can establish those products which are indigenously already been developed into a formulation, and that can be also be done. So there are multiple options that are available, one can utilize their own way. So how can we improvise this process? So likewise, when we have the large population and India being where under reporting of these adverse events compared to the other, because the PVPI pharmacovigilance program of India, where we see very minimal reports for everything. It doesn't mean that the individuals are not facing the safety concerns for the medicines that they have taken, it is not being reported. But that is the reason it's most important that it has to be reported. So the question arises is, if we keep reporting this big population, how it can be managed? And there the AI can help us, because whenever the case is inflow from to the database, automatically from the dashboards, whatever the cases, because the cases can be reported from the market, that is spontaneous cases, or from the clinical trial, or from the literature. So whatever you report in the literature or published literature, those also come into the database. So that's the reason whatever case scenario that we have seen, like sir has shown one case study report of a mother facing with a. Similarly, one can also, whatever in locally, whatever you see in this scenario in your relatives, family or anything, that need to be published, so that that information will go into the database as a safety concern. Because completely evaluating all safety concerns of one product will lead to a signal. Unless, so the every product that AI, what it is doing is, we are using this literature cases. In a week, we receive around 10 to 20,000 of literature cases. So there may be valid cases, there may be invalid cases. So when we inflow through this AI, it will automatically, because we have the algorithm developed, and with that automatically it will deliver what are the valid cases list, and these are the invalid cases list. So invalid cases list can be thoroughly examined, because we need 4 minimum criteria: the patient, the reporter, the event, and medicine. Without medicine, the event cannot occur. So with these 4 minimum criteria, one can report. Without these 4 minimum criteria, you can't report any adverse event. So with this information, the AI will assist you in developing a fast validation of valid and invalid, and also there are some, means it is 80% accurate, where from the flow it can be directly incorporated into the safety database, and it automatically narrate the story of what happened to the patient. That is called the medical history, and from that this will be useful. So from that again we will be reporting, developing into an again reports, aggregate reports that we mention as PEDARs for US FDA, PSURs for the other regions, periodic safety update reports. Those need to be updated. So these AI, when we are using this, around 70 to 80% the aggregate reports can be generated, another 20% at this moment. But moving further, these reports can be fully automation once we have the data and generation. So those type of concepts can be developed in the and similarly in the other real world evidence studies, because clinical trials are controlled studies where with minimum information, definitive inclusion exclusion criteria. So we have one type of data or a sample of data. Whereas when it leads to the population, it may not be an appropriate. Hence, most of these products are eliciting adverse events in the phase 4 or post marketing. So hence, these real world evidence studies also became very mandatory, or that also can be focused on. So these are the various evidences in pharmacovigilance also, like we can go into and these are the various opportunities. First one being the futuristic personalized medicine or precision oncology or real world evidence or repurposing or streamlining the clinical trials, including the pharmacovigilance. Saying this, there are multiple problems. Definitely there will be a mitigation for everything. So one can focus on this. And finally, in the best interest of the time, so this healthcare, there are much more research, promising molecules are required. That as sir is instructed, it is you from the precision medicine or from the stem cell therapies or whatever. That is the one thing which can be focused from now onwards, so that you can become excel by the end of your graduations. And also on the safety concepts, it's like an IT job where you can sit on the system and whatever the adverse event that comes to you. And in my team, around 80% of them are females, so that it is easy for them to make their career opportunity easier. Unlike clinical trial where you have to travel to the clinical trial sites. So that is the so just I am giving a brief outline on the what AI can help us and how we have to. So AI will not replace the professionals, but professionals without AI knowledge may not be successful in, going forward. So please understand this concept. And if any questions, as it's a lunch time, I'm sorry, but each one can reduce the sufferings and save thousands of patient lives reporting the adverse events. This is the only punch word which I am giving. So first thing is this, have this in your mind. So keep reporting as a professionals. And the second thing is always have the information given by respected seniors, that you should have the main criteria is ask, ask, please interact. It is not that easy to get such big professionals here. ASK means attitude, having a positive attitude, you will have a career opportunities. Second one is skills, soft skills that you have to develop. And third one is knowledge on the profession or whatever the area of. So saying this, I thank you again the VIPER management, Ramesh Garu. So thank you everyone. Thank you.
We extend our sincere thanks and gratitude to our chief guest, Dr. Krishna Devarkonda, and our guest of honors, Dr. Ranga Namburi and Dr. Sridhar, for their valuable time and traveling all the way from city, long distance, and only thing to educate all the young and sparkling minds. Thank you, sir, for your patience and everything. Taken time for us. We acknowledge the presence of various principals from various colleges. We thank you for all the principals for joining us. A special thanks for all the delegates, as at 829 delegates who have joined online and offline. Thank you for joining us. Thanks for the faith. Hope you enjoy and take the treasure what is suggestions given by the experts, which will carry through the lifetime, I believe. Special thanks to the admin team, BVRIT and VIPER, and electrician and sound system, everyone who are working behind. A special thanks to our VIPER team who has made this all event a great success. Thank you all. Thank you all for your patience. Right. Thank you.
We are now done with today's inaugurational session keynote address here. Thank you all for your gracious presence. Kindly note the buses have been arranged on the left side for the conference. Thank you.
Yeah, delegates. Buses are arranged to the left side. If you go to the left side, buses are arranged. They will take you to VIPER. The sessions are end here. From now, all the sessions will be carried in VIPER. From tomorrow you can directly come to VIPER.